Okay, good. So let's start. I want to welcome you all to this 11th CNAG symposium on genome research. This time we have picked cancer as a topic because we've done that before. But I will explain to you why we picked cancer. It's a real delight to have you all here in person. It's different than what we're used to from the last couple of years, but I think we also should get used to this again and seeing people face to face, because the interaction that you get when you talk to someone is just so much better than when you're on a video camera and everything is two-dimensional. So I'm absolutely delighted that you're all here and that so many of you decided that you would come here and be with us for the day. Now, the objective why we do these symposia is essentially to bring some of the work that we do with our collaborators to everyone, so that everyone can see what it is that we actually do with the different collaborators. And what I should say is that we run on the order of about 400 projects per year. And people sometimes don't realize how many projects we actually run. We have about 200 distinct individual PIs who are collaborating with us. So on average, about 2 projects per PI in one given year. And this is all managed with a lot of infrastructure and a lot of organization and a lot of making sure that we are working up to very, very high standards and so on. So I just historically want to go back a little bit and tell you something about CNAG. So we were created now nearly 13 years ago. We were put in this science park here. And we have funding coming from the Ministry of Research in Madrid and from the two ministries here in the Generalitat. We do collaborative work with people. So the idea is we do not want to replace BGI. We do not want to just receive a piece of DNA and give you back FASTQs. That's not the idea. We want to work with you on your research project. So we will try to understand what it is that you are trying to achieve with your project, and we will try to do the best to take that all the way through to the results. And that in some instances even means that there is an analyst who will actually tell you what the result is. So we'll say, I think the result is gene X. And goes all the way down to even helping with writing materials and methods for paper and giving advice. And we think this paper would be better in this journal over the other journal. We had a conversation like that yesterday evening over dinner. And so we try to really work with you. And that makes us different than BGI. We have a couple of things in our mission. The mission is that we are meant to carry out large-scale projects in genome analysis in collaboration with the community. Now, we crossed out that large scale after a few years. The reason for this was that 13 years ago, to be large scale, you had to sort of put 10 machines on the ground, and that generated 1 gigabase per day or something like that when you ran all of these machines. But nowadays, the machines are so powerful that essentially every fraction of a machine run is way more than what we did in a day 13 years ago. So we do with everything that we had then. So the large scale we just took away, because essentially any experiment you do in genomics nowadays simply is large scale. Then in terms of how do we try to do things? We try to do things up to a very, very high standard. So this is important. We don't want to be just giving you anything. So the idea is really do everything up to a high standard. So we have SOPs in place, we have ISO standards in place, and we follow these SOPs really down to the T. So it's really, you do exome sequencing, and that really means you do this quality check, this number has to come out, this has to happen here. So all of this is very, very well structured. So quality is sort of nearly the top parameter that we look for. It has to be good, because essentially the story in all of this is, if your quality is good, you can make a good result out of it. If your quality is not good, then you have trouble identifying your result, and then sometimes you publish things that then end up being wrong because you just ran after an artifact that came out of the way you did your work. So quality is the most important thing. Now, over the years, we've installed what we called a genome engine. Initially, this looked really quite impressive because we had 11 or 12 genome analyzers standing around the wall like that, and they really made a semicircle. Nowadays, because these Illumina NovaSeq instruments are so powerful, we don't need quite as many any longer. So we don't have 12 of them as we used to have then, but we have 3 of them at the moment, and we're probably going to get some more. We've diversified. We have other sequencing instruments as well, so we now also do quite a lot of work on this nanopore sequencer here. And obviously, a lot of the activities we do have sort of been sort of extended. So we go into other activities and do other protocols as well. I will show you some of that. And then the whole thing is backed up with a very powerful computer. And we actually noticed that we have a computer that is a measurable fraction of what the BSc has. It's just up the road. So we are not 0 point something %. We are like 5 or 10% of what the BSc is in terms of compute. So it's actually an awful lot of compute in our own right. And we actually need it and use it a lot. Okay, I said that quality was very important, and we underline what we do with ISO standards. So we've got an ISO 9001 certification, and we run the major applications under an ISO 17025 accreditation. This is an accreditation that allows you to take your results and feed them into clinical work. So you can give this to a hospital, and you can say, this is the result, and you can use that to establish a diagnosis for a patient that you have in front of yourself. We're working on getting 15189, which is the immediate clinical accreditation. There's a technical problem that we have to solve with that, which is how we are structured economically. But once we have that solved, we're going to go for 15189, which means we will be able to deliver these kinds of analyses, whole genome sequencing, exome sequencing, RNA sequencing, straight to the user. So a clinician who sort of says, I need that for the diagnosis of my patient, can come directly to us and ask for that. Strategic areas that we do. So we have essentially 6 boxes that we work along. So we have personalized medicine, which is turning into a major chunk. So much of what we do now moves closer to the clinic. So it's delivering exome sequencing, whole genome sequencing for rare disease analytics into clinics. And here in personalized medicine, we have a huge initiative in Spain, which is called IMPACT, with multiple projects behind it, that is working towards implementing genome analysis into clinical care. Rare diseases is something where we've done a lot of efforts over the years. We've built systems, analytical systems that allow bringing the data that is obviously very complex because it is very big, but allows you to bring it very, very close to the user being a clinician who has a patient sitting in front of him, and with clicks in a system can basically figure out what a potentially pathogenic variant is that is causing the disease that the patient sitting in front of this clinician is suffering from. Cancer, that was the reason we were brought here, and Elias, I'm really delighted that Elias is going to be the first speaker in a minute, and he's going to tell the story of how we started, because essentially CNAG was established to support the ICGC CLL project that Elias headed up, and there's been so many things that have gone on after that, and I'm sure he's going to tell us the entire story of what we've done in that, all the way to the kinds of analyses that we do nowadays for cancer. Now, in cancer, we do many projects as well, so we extended our activity in cancer vastly over the years. About 8 years ago, we decided that single-cell analysis was a very, very important topic. So we started investing into single-cell analysis and established an entire section, a team that is dedicated to single-cell analysis. This is headed by Holger Hein unfortunately, Holger is sick today, so he will not be presenting. He's on the program. I know he's one of the stars on the program, but unfortunately, he's sick. He doesn't have COVID, I think, but other infectious diseases put people down these days as well. So that's unfortunate. We will think about how we will, we know that we can't substitute him, so we will think about something different to do. We'll have a community game that we'll all play together instead of that in that half hour. No, we'll do something else. Then an activity that we call the genome in action is more or less epigenetics. How does the genome actually work when it is doing its activity? So how does the nucleus reshape itself? What are the marks that we find on the DNA? How does it move? What does it result in, and so on? That we call the genome in action. We have quite a lot of activity in that, and we have a new high-resolution imaging system that is going to come into the institute in probably about 3 months' time. So this is a system that allows looking at the nucleus really as it is sort of winding itself around and doing its work. And then recently we started into biodiversity, very important, obviously not directly disease-related, but we look at doing de novo assembles of species that don't have a reference sequence. We have a lot of expertise in this, and we put this towards the activity that is going around Europe. And we have one project that is starting very, very shortly, which is called the Biogenomics, Biodiversity Genomics Europe, BGE, that is going to put up about 500 new de novo assembles of species of different levels of interest, so endangered species, economically interesting species, and so on. Okay, now you've noticed this is the 11th one of these symposia, so Anna made me a list of all of the symposia that we've had, and I thought I would just run you through this. So there's been 10 before. The first ones were very heterogeneous. We basically touched all of our boxes, and then as we moved along, we became focused. So we started running the symposia on specific topics. So we had ones on rare diseases. We even did one in Madrid. So we did a symposium in Madrid on rare diseases, but sponsored by us. Then we had ones that were dedicated to agrogenomics, with speakers that are interacting with us on that level. We had personalized medicine that we did a few years ago. Then we came back to rare diseases, again went to Madrid for a presentation on that. Then we did this genome in action thing, and then last year we did a symposium that we did online. I don't actually know how many people participated when we did it online, but I think the number was quite remarkable. Which takes us to today's symposium. So this is the program. As I said, Holger, who would have been just before lunch, unfortunately can't be with us, and then we have another cancellation just after lunch, which is Marianka Schmidt, also infectious disease, also not COVID, and unfortunately couldn't travel. So this is the reality of what we have. We've all gotten our immunity a little bit suppressed over the last couple of years, and haven't worried about any other bugs than the one that obviously is the elephant in the room, and these other bugs now hit us, and we get sick of those. Okay, and with that I will get to the acknowledgements, and today I want to acknowledge the sponsorship of Illumina. Illumina is sponsoring this, and Illumina is taping in a presentation. It comes online afterwards. There's going to be a presentation that is going to be given by someone from Illumina. I've forgotten where it was in the program, but there will be that, and it is a topic that is also one that we are picking up during the meeting today. Okay, with that I'm gonna stop, and I'm gonna hand it over to Elias. It's a huge delight to have Elias talk to us here. Good. I don't know where your presentation is. Do you know where your presentation is?
Yeah. This one.
Okay, perfect. Great.
I saw it was in the first one.
Okay, good.
Good morning, and first of all, thank you, Ivo, for putting together again a program and bringing us together face-to-face, an activity that is essential to communicate science. The Zoom is okay, but not real life. And thank you also for the fruitful collaboration of more than 10 years now of putting together this integrated view of how genomic studies can illuminate our clinical practice in diseases and how we are integrating this knowledge in changing our clinical practice. And we have been doing that with the CNAG team now for more than 10 years when we started in the ICGC project with sequencing the first genomes of chronic lymphocytic leukemia 10 years ago. And as you will see, the story continues in different pathways. This disease is the most common leukemia that we have in the Western world in adults, and it's a disease that we know very well the story. It starts with what we call pre-leukemic phase, monoclonal B-cell lymphocytosis. The disease becomes a leukemia, but in most of the patients remains stable for many years. In some patients, and still for reasons that we don't know well, starts to progress. The patient requires treatments. These treatments do relatively well, and the disease regresses. In some of these patients, after several lines of treatment, becomes refractory to this disease. And unfortunately, in 5 to 10% of the patients transform into an extremely very aggressive lymphoma that we call diffuse large B-cell lymphoma, that we call, you will see why, Richter transformation. As Ivo said, we started 12 years ago to sequence the first genomes. And within the ICGC, we were the first consortium to sequence the goal, that was to sequence the genome of 500 patients. And this is what we call the theater of this perspective of the genome, in which each line corresponds to a patient, and each row corresponds to a driver alterations that we consider at that time. And this image put together by Josep in Oviedo, I think is a nice shot of what happened in this disease. You can see that all the drivers are concentrating in 2/3 of the patients, and we have more than 1/3 in which they have only deletion in chromosome 13, or even there is a group of patients, 15% of the patients, that we don't, that time we didn't find any driver. But the surprise was that at the same, more or less at the same time, the colleagues of Dana-Farber and Broad Institute also sequenced around 500 cases. And the surprise was that when we put together the list of drivers, you see that we only have 40% in common. There were more than 60% of drivers that were different in both series. And these 50% of the patients in both series that didn't have drivers. So that tells us already at that time that this, in spite of this massive effort, we have not complete understanding the genome alterations in CLL. That's why from competitors, we become collaborators with the Broad Institute and Dana-Farber, and put together our series, and also we sequence some more additional cases, coming with a series of 1,100 patients in which the whole genome or the whole exome have been sequenced. Though the pink cases are the ones from the Spanish consortium, and the different colors correspond to different participants in this massive effort. Also, in addition to the genome, we had the transcriptome, epigenome methylation, etc. We just a few months ago, we finished this massive analysis in which we identified novel drivers. Intriguingly, in this disease, and tells you that tremendous heterogeneity, most of these drivers are present only in 1 or 2% of the patients. And 24% of the patients have at least one novel driver. And we move from 50% of the patients without drivers to only 4% of the patients still without null drivers. But I want to emphasize that the discovery of novel drivers require novel technologies to interpret the genome. And that's why in this and previous studies, we have identified novel drivers that were require novel pipelines. And one of them was a mutation in the immunoglobulin receptor of the CLL. That is a particular mutation, and that was only possible to be detected in the genomes thanks to a novel pipeline that was developed by Ferran Nadeu in our group. But also, there was a mutation in the small RNA non-coding of the spliceosome complex U1 that was discovered, that is in a repetitive region, was only discovered thanks to a new pipeline developed by Lincolnstein in Toronto. And also other mutations were discovered because there were also with new pipelines taking into consideration the three-dimensional structure of the proteins by pipelines developed in the world. With these novel drivers, we have expanded the number of pathways that might be altered in this disease. We are not going to go into the details, but gives you the complexity of the molecular alterations in this apparently simple disease. But also the transcriptome analysis gave us new clues. We identified 8 clusters of patients, clusters of transcriptome groups that identified, that separate the 1,100 patients. And each of these clusters were associated with particular mutations. And what is important is also these clusters split the prediction of the outcome of these patients quite nicely. So it's an idea that we need to integrate genome and transcriptome information to understand better this disease. However, all these studies have been virtually all these have been performed at the moment of the diagnosis of the disease. As I told you, the evolution of the disease is long with many different ups and downs, and this phenomenon of transformation that we call pathologies, we call Richter, because Maurice Richter was the pathologist that almost 100 years ago described, and in this original paper with these images that were drawings. And we know well that in the clinics, and we know that this is a very dramatic situation because we don't have therapy for these patients, and when the CLL transform, the patient survival is less than 1 year. But what are the mechanisms leading to this transformation are not well known. And that's what we tackle again, this study together with many people, as you will see, in which we took advantage of our biobank, in which we have 19 patients in which developed the CLL, developed Richter in periods of time that were from the very moment of the diagnosis. We simultaneously diagnosed both the CLL and the transformation, but in other, the transformation occurred up to 19 years after the moment of diagnosis. And we had samples all through this evolution of this patient. And you will see how important it is to have these multiple samples. And we have performed a comprehensive analysis that goes from bulk genome sequencing, epigenome, transcriptome, to high resolution, including single cell analysis. Those are the patients, and you see in each patient we have multiple samples and different treatments. Those who were at diagnosis, those with regular chemoimmunotherapy, and those that included new treatments like inhibitors of the B-cell receptor or inhibitors of apoptosis like venetoclax. And we perform whole genome sequence in virtually all patients. And in addition, in 5 of these patients, we were able to study deeply different layers of epigenome alterations and single-cell analysis together with transcriptome. And those are the main results. And the genome analysis were performed by Ferran Nadeu and Romina Arroyo at the BSc. And we found mutated genes in cell cycle alterations with novel findings like translocations of cyclin D3, mutations in cyclin D2. We also observe alterations in MYC, translocations, amplifications, even in MYC-CN. Mutations in Notch, that we were the first to discover that Notch mutations were associated with these translocations. We expanded that not only Notch, also mutations and alterations in genes of this pathway. Also alterations in NF-κB elements, and tremendous alterations in the chromosomal complexity, including chromoplexia and chromotripsis. Sometimes it's a challenge how we interpret all these multiple individual alterations, individual genes. But I think when we put together this data, some idea starts to appear. You can see that in virtually all patients, we had an alteration in a cell cycle regulator, and that fits well with the tremendous highly proliferation of these tumors. Also, we found alterations in MYC in virtually all patients, except a group of patients, but those patients have alterations in Notch pathway, and we know that MYC is a downstream element of a Notch activation pathway. So if you see, virtually all patients had alterations in MYC/Notch. But MYC, in addition to create proliferation and cell growth, also is a potent signal to induce apoptosis. So the cells, to survive, require to activate survival pathways. And this is what we see here with the activation of NF-κB pathway that is present in virtually all cases. So all this mare magnum of single cell alterations, when we see integrated, I think a picture of pathogenesis comes out in a more clear way. But in addition to the mutations, we also observe a high number of structural variant alterations that are not present in CLL, chromotripsis, chromoplexia. But I want you to look at these images because I come back later. Some of these chromotripsis alterations were targeting several of the drivers at the same time, and I will come back to that later. And looking at the mutations, was interesting to see that the CLLs that in this study are circles were mixed with the other CLLs, conventional CLLs that we had. But when we look at the Richter in the principal component analysis, the Richter samples had a totally different apparent mutational profile. And that's why Romina and Ferran looked more carefully at to draw the mutational signatures in these cases. And we came with some signatures that were mutational signatures that were well known, and we'll come back later to that. But also we discovered two new signatures that have not been previously described in cancer. And one of them, we call it Richter signature, because we didn't know what that signature was and was not previously described. And another signature that had not been described, but where we were doing the project, it turned to be that this signature was associated with the treatment of the patients with gangliosiklovir. That is a treatment for cytomegalovirus infection. And when we back to the history of this patient had received previously to the Richter this treatment. So we started to think that the genome of these patients at Richter might carry the imprint of previous treatments. And that was important for a couple of reasons that I think is relevant for the study. Look at this in red, the signal of this signature that we discovered in the Richter sample, but we saw that this was also present in previous samples of the CLL before transformation and even years before. Other signatures, I will not go into details, were previously described, but to try to understand what this new signature was, we put together each patient in each line together with the different treatments that these patients have been received. And you see that the signature with that I mentioned, gangliosiklovir, was in the only patient that have received this treatment. We also detected a signature that had recently described on melphalan treatment, and it turned to be that these two of these three patients have received melphalan. So we thought that this new signature could be related to a treatment, and that was the case. Look that all the patients that have this signature in the Richter had received chlorambucil or bendamustine in the previous years. But what I think is important in this signature is that this introduction of these mutations in the genome is random. So to see this signature in a sample means that this sample comes from the expansion of a single cell that has a particular signature in the genome. If we go back here, that means that what we imagine is that the sample that had received this signature was expanding clonally before the Richter transformation, many years before. And that's what Ferran and Romina could confirm by reconstructing the phylogeny of this clonal expansion, in which we saw that these clones could be predicted to be already present many years before, in this patient, 14 years before transformation, already at very beginning of the disease. But also the second point was that this Richter's signature was present in 2 different clones, predicted to be. It turned to be that in this particular patient, we have cells still in our freezers from different time points. So we went back to look at the flow cytometry of these patients, and in time 4, that was after the patient had received bendamustine, we already detected 2 clones of 2 subclones of the CLL with different markers. And one of them was, the cells were larger. At time 5, the 2 clones that is here, the 2 clones were present, but also a clone here was appearing. And at the moment of the Richter transformation, we have the Richter, that is this one, and this is pink clear subclone, that is this one, had disappeared. So this prediction of the whole genome, we could confirm that by flow cytometry that was occurring here. But that was telling us that probably this going back could be real, not just an in silico prediction of these subclones. So that was the picture. We have 5 patients in which we could detect this, or we could predict this early beginning of the Richter transformation many years, 19 years, 14 years, 9 years before the actual Richter transformation. To confirm that, we move a scale in our analysis, and single cell with only points can give you a beautiful picture, a more clear picture of what could be happening in a tumor. And this is another patient. You see, from diagnosis to Richter transformation, 14 years, we have multiple samples. The Richter sample had a P53 mutation that was not present in the CLL before. In these clones that come from only one, carry another different P53 mutations. By doing a single-cell analysis of this panel of 34 genes, we detected that the clone carrying the mutation of this mutation of P53 was already present at different time points of the disease, including 0-1 point cells 14 years before the transformation. And this clone that had this other mutation of P53 at time 3 and 4 also had this mutation already at the moment of diagnosis 14 years before. So we were detecting the mutations that gave rise to different relapses and the final transformation 14 years before that happened. We wanted to go a step forward and confirm that if that was only mutations or also the transcriptome profile of the Richter cells. And that's what the group of Holger Heijn, Ramon, mainly Ramon, but others, look at the single-cell transcriptome of all the samples of this patient and detected a particular profile of CLL that I will not go into details, and also a profile of the Richter clearly different. When we look individually at the samples, you could see that still at the Richter we could detect the CLL cells, but those are the Richters. But important is when we go back in time, up to 14 years before, the samples contain or could be identified samples with the cells with the transcriptome of the Richter already at all time points, including at diagnosis 14 years before. And the question is only the transcriptome, is only single mutations, or also that chromotriptic alteration that I show you that some of the Richters have. And Ramon used a pipeline that could infer the chromosomal configuration by analyzing the coordinated expression of the genes that are in a particular region of the chromosome. And what we saw at the Richter is that the expression of a series of genes in these chromosomal regions that were deleted in the chromotriptic phenomenon were clearly downregulated in the Richter sample. But what was very interesting is that the samples before the Richter, up to 14 years before, there were cells that already carry this pattern of downregulation of genes that allow us to think that this chromatoptic phenomenon had already occurred 14 years before at the moment of diagnosis. The confluence of all these alterations made us think that those cells had acquired mutations, transcriptome, and complex chromosomal alterations already at the moment of diagnosis and were silent, dormant for many years before the different relapses. And we look at the epigenomic layers by Bea Garcia and Iñaki because we look at the methylation, and the methylation didn't give us many information. The profile of methylation of the Richter and the respective CLL was not very major differences. However, in the Eastern mark activation and active regulatory regions, open chromatin accessibility and expression clearly show us that the Richter had a different transcriptome and regulation of the transcriptome, different epigenome transcriptome. When we look at the comparison of Richter samples with the Eastern marks and open chromatin compared to the CLL samples and with normal B cells, different types of normal B cells, clearly the Richter cells have a different modulation of the epigenome. And in these regions that were open de novo in the Richters, the analysis of the motifs for transcription factors identified that the most significant were TAD4, that we learned at that moment that is related to control several pathways of metabolism, particularly phosphorylated and oxidative, also MAS related to MYC, A2F related to proliferation. So it started at the epigenome level. Makes sense what we had seen before with the mutational and transcriptome. And when we look at the RNA-seq, the differentially expressed genes in the Richter transform also were genes related to these pathways. And when we look at the pathways, very interesting, several pathways became upregulated in the Richter cells. I want to emphasize MYC, cell cycle, and I want to highlight phosphorylation oxidative. But also, surprisingly, the B-cell receptor pathway was downregulated. And we confirmed that. And also, when we look at the single cell, Ramon derived a score that we showed that the OXPHOS was high in the Richter sample, but also in the initial samples in those cells that we have identified also have this upregulation of the phosphorylation oxidative and also have this unregulation of the B-cell receptor. And very important, these Richters have occurred after treatment with ibrutinib, that is inhibitor of the B-cell receptor pathway. So now we start to think why these patients develop Richter, because these cells are not sensitive. They don't need the B-cell receptor pathway because it's downregulated. So if you do an inhibitor of the B-cell receptor, the other cells will be inhibited, but those don't. And that will be a signal that may allow these cells to proliferate. But again, this is in silico studies. Can you demonstrate molecularly, biochemical, that this is happened? Again, we had frozen cells, cryopreserved cells of these samples, and thanks to the collaboration of Heriberto Barrera in the group of Dolors Colomé at IDIBAPS and Pablo Garcia at IDIBAPS, we showed that the phosphorylation oxidative of CLL in Richter and the corresponding CLL was significantly higher in these two patients. This is a different pathway that is interesting. And also the B-cell receptor that we signaled that we could study by the flux of calcium was down in the Richter sample compared to the respective CLL. So those predictions of the bioinformatic studies were confirmed biochemically. But then the important thing is that OXPHOS pathway might be targetable in the clinical setting. So we inhibit the cells in vitro, the phosphorylation oxidative. Nothing happened in the CLL, but within the Richter sample, we give this inhibitor of the complex 1 in the mitochondria the proliferation of these cells stops. So we have discovered, you see the reduction. And this is not only in our samples, this is also, time is over, but I have finished. There is one case of Richter syndrome published at single-cell level in the literature by the Cafibu in Dana-Farber, is this patient. And Ramon looked at the score of the OXPHOS and B cell receptor, and you can see that also in this patient that is not from our cohort, the OXPHOS is up, the B cell receptor is down, suggesting that this is not just our finding, is also in another Richter that was demonstrated in others. So, in conclusion, we have traveled a lot since we started with FIBO and the CNAG and the people in Oviedo sequencing these genomes 10 years ago. But now I think that 10 years later, we have virtually completed probably the map of drivers in CLL at the moment of diagnosis. Still, we don't know, as you have seen, what happened in the evolution of these patients. This transformation of CLL into a very aggressive lymphoma years later is configured by the integration of alterations in cell cycle, MYC-Notch pathway, and chromatin remodelers. I think it's very interesting to me is that the CLL carries these imprints of the treatment that we give of the patients and is in the genome. And after analyzing these imprints of the treatment, we are starting to understand some steps in the evolution, as it is the selection of single clones that give rise to the different evolution afterwards. And this transformation and history, at me, of the evolution of the CLL, it seems that is already written at the first moment of the disease by a massive diversification of the tumor clone in multiple clones that later on will give rise to different relapses and transformation. And this pathway, OXPHOS and BCR, could be, seems essential or important in this transformation and could be targets of treatment. I think we never emphasize enough how many people participate in these studies. And I think all of them in different aspects are essential to develop these comprehensive studies, but I already mentioned key people like Ferran Nadeu, Heribert Play, and Beatriz Garcia in our IDIBAPS, but also Romina Arroyo at the BSc, Ramon and Holger here at the CNAG, but this is an interesting adventure in the synergy that we are doing. And Iñaki in Oviedo, Josep Puente, and the group at Dana-Farber. And this is founded by La Caixa Foundation and our ERC synergy with Ivo, Iñaki, and Holger. Thank you very much for your attention.
Thanks a lot, Elias. Questions? Please.
Just a second.
Oh, thank you. You mentioned chromothripsis as part of or a feature for CLL and Richter. But do you think chromothripsis is coming along with the disease, or it's actually the driver of the disease? I mean, you also show that there were genes down-regulated or up-regulated, but is it the driver of the disease? That's my first question. And my second question, is it always occurring on the same chromosome or even on the same sites of the chromosome?
To define drivers is a story that I am not going to go into. What I can say is that in the different chromothripsis phenomenon that we have seen in different cases, in virtually all cases, involves some genes that we have defined as drivers. because they are altered by mutations, by deletions, or by translocations in other cases without the chromotriptych. So definitely I think that chromotriptych phenomenon targets drivers that will be relevant for the disease. That's something that is there. And also what we have shown is that this phenomenon is occurring very early. Not always is the same chromosomes. In different cases are different chromosomes and different genes targeted by this. So it's a phenomenon that occurs, and my idea is that targets different genes that might be really drivers of the disease.
Because we have observed the same with osteosarcoma, even with a higher number than previously reported, that chromotripsis is present in up to 70 or 75% of osteosarcomas. And there may be drivers in it, but the chromosomes, they are not always the same, and we are not sure what we are looking at.
Well, I think the picture comes more clear when you have many points in the picture, and that's what I think, the fact that's what I wanted to summarize all these alterations in different individual genes in these pathways, because when you put this graph, it seems that all patients, all cases have alterations by one gene or the other, by one mechanism, translocation, mutations, chromotripsis in these genes, but different phenomena, but all converging to these genes. And when you put together all the cases, virtually have alterations in these 4 pathways. That's what I think is, to me, is relevant coming out from this study.
Elias,
Jordi.
thank you very much and congratulations as usual. It was a fantastic talk. I have a question from the clinical standpoint about the way we should follow these patients in the future, because now there is a widespread use of very effective drugs, BCR inhibitors, BCL2 inhibitors, and but after some years these patients can progress and we can lose control. And I was wondering if we should monitor, we look for search for these subtle subclones that can anticipate the progression and perhaps intervene in a, it's a different way of.
Definitely, I mean, coming from the clinics, this is the question that we are wanting to address. We are fortunate that this is the first study in the clinical setting that we are doing in this synergy program together with Ivo and Holger here, an amazing group of young people in the bioinformatics world. That is, I was telling Sergio that I love the seminars on Friday because it's a learning experience. So now we are targeting the CLLs that in different pattern of evolutions. We took the Richter because it's the more dramatic, but we have patients with CLL that after these treatments relapse after many years. Why they relapse? Why they stay in response for so many years? So now we have a number of samples in which we have this sequence in patients that have relapsed after years, that are resistant to these therapies from the beginning. Even we have some samples in which the CLL disappears spontaneously with no treatment. So we are starting to analyze that from the same perspective. We have already done the whole genome and transcriptome and histone marks in some of these patients, and now we are starting to look at the single cell. Yes, I think there are clues that are very interesting in the clinics, because I didn't mention, but these patients that were treated with ibrutinib, by doing the B cell receptor analysis at deep sequence and at single cell sequence, we detect mutations that are again also present at the moment of diagnosis that generate a truncated B cell receptor. That means this cell, the B cell receptor, doesn't work. So to survive, it has to develop other survival pathways. Well, this clone, this cell that we can detect, we detected one cell out of 100,000 at the moment of diagnosis. After the ibrutinib treatment, increased to 10% of the tumor cells. So clearly, this subclone that was present, I again said out of 100,000 has increased to 10% after the ibrutinib treatment. So I am starting to think if we are able to detect these inactivating mutations in the B cell receptor, or we can detect at the single cell level the B cell receptor downregulation in some of those clones. I don't say we should treat the patients with ibrutinib because it's extremely effective, just that we have a tool to monitor the evolution of the disease and decide if we stop or change to another drug. Definitely, Sergio was asking me if we will introduce single cell in the clinics. I am sure we'll introduce it in a few years.
Hi, thank you, great talk. And I was wondering, out of curiosity, the cases that have the P13 deletion, how do they fit into all of this?
The P what?
The P13 deletion, so the CLL cases, at the very beginning you showed the different types of...
Alteration?
Yes. So in this study they are not, for this study they're not part of this, so there were no cases that had this deletion.
I don't know, P which?
The P30 deletion?
P30?
13, sorry, 13, sorry.
Chromosome 13.
Yes.
Oh, yeah. Chromosome 13 is the most frequent chromosomal
Yes,
alteration.
that's why I was asking about it.
Yes, and the idea is that the MIR 15 and 17 in this chromosome is the target of this deletion. And it's postulated that this is targeting the BCL2 pathway by increasing the stability of BCL2, so giving kind of survival resistant survival advantage to the cells carrying this deletion. So
okay, so there's an entirely different phenotype.
No. I mean, the CLL cells with 13 probably is one of the earliest events in this transformation, in this CLL disease. Okay. 13q deletion. Actually, we detected this 13q deletion in this pre-leukemic phase that I mentioned, the monoclonal B-cell lymphocytosis. So it's relevant for the disease.
Thank you.
Hi, Elias. Very nice presentation. I have a question about one specific pathway, which is immune escape alterations. So in diffuse B-cell lymphoma, it's very frequent that we observe B2M loss, CD58, and so on. Whereas for CLL, it's rather infrequent. So what about this Richet syndrome?
I think it's very important question, and this is one of the findings that in these diffuse large B-cell lymphomas, although morphologically, phenotypically are relatively similar to conventional de novo diffuse large B-cell lymphoma, the profile of mutations are different. And this is one of the aspects that doesn't come out as a major targets in this transformation. We don't see deletions or inactivation of beta-2 microglobulin. We don't see CD58. We don't see CD70 that are common immunocheckpoints targets in common diffuse large B-cell lymphoma. It's also contradictory, and perhaps Jordi can correct me, contradictory results treating with immunocheckpoints inhibitors in Richter. Definitely in CLL don't work well. There is some, to me, still not clear results in the CLL, in the Richter. Probably the lack of these mutations in these cases may be, but there is a player here that also is the target that we are going to study in the new Leukaemia project that we just got funded. Here we have been talking all the time about the tumor cells, but the tumor don't live alone. They live in the tremendous complex microenvironment that needs to be integrated in the picture. And I think this is one of the another steps in understanding the evolution of the disease.
Thank you.
With the goal of modulating. Thank you so much.
Good. It's my pleasure to welcome the next speaker. This is Talia Velasco from the Josep Carreras Leukemia Foundation. Research Institute. So I will not tell you how I told her she should picture all of us while she's giving her presentation.
Okay. So first of all, I would like to thank the organizers for giving us the opportunity to present the work we have been doing in the lab for the last couple of years. So obviously, we are interested in leukemia, and in the lab, we are studying different kinds of leukemia. For this specific project that I'm going to present today, we chose to study the acute myeloid leukemia, or AML. This kind of leukemia is characterized by a block in the differentiation in the myeloid lineage and a clonal expansion, which accumulates a lot of myeloid blasts in blood and bone marrow of the patients. But what interested us more of this disease is that this disease follows the theory of the cancer stem cell model. So the cells of this disease are organized in a hierarchy with a subset of cells in the top of this hierarchy known as leukemic stem cells or leukemic initiating cells that has some special properties specific for the stem cells, like in the healthy tissues, that through a differentiation process give rise to the rest of the blast cells of the disease. And these properties, one of these properties is the quiescence of the dormant status that these cells are in. And this has specific and important implications for therapy, since the chemotherapy drugs and chemicals that are used in clinic for treating these patients specifically target the more rapidly dividing cells, that the blast of the disease, but not these leukemic stem cells, which make them more resistant and survive to the treatment and give rise to the disease months of years later after the treatment, producing the relapse of the patients. So one rationale to be able to target these cells and to avoid the relapse of the patients is to activate them, to make them cycle. So in this way, these cells could be sensitized to the chemotherapy and eliminated. And in this case, we can achieve the long-term cure of the patients, avoiding the relapse later on. So to exploit this mechanism to avoid the relapse of the patients, first we have to study which signals or which mechanisms keeps these cells in this quiescence or dormant status. And here I'm showing you just a brief image of all the signals that the normal HSCs, the hematopoietic stem cells, that are the healthy counterparts of the leukemic stem cells, receive in the niche where they are located, in this case the bone marrow niche. And you can see many different signals, cell-cell interactions or soluble factors. But the one we were interested in, and the one I'm going to tell you a little bit today, is the low oxygen concentration or the hypoxia status that exists within this niche. And how the cells can sense these low oxygen concentrations within the microenvironment. So exists a family of transcription factors known as hypoxia-inducible factors, or HIFs, that its regulation is controlled at the level of protein by the levels of oxygen. So in presence of oxygen, in normoxic conditions, the oxygen in combination with the PhD enzymes can hydroxylate different resites of the alpha subunit. Now, the von Hippel-Lindau protein, or VHL protein, can recognize these hydroxylated resites and promote the ubiquitination of this alpha subunit and promote its degradation. However, when the cells are in hypoxic conditions or there is no oxygen available, the alpha subunit is not degrading, it is accumulated in the cytoplasm, which can be translocated to the nucleus, dimerized with the beta subunit, and form this transcription factor that recognize the hypoxia response element in the promoter of many different genes, activating many different genes participating in processes such as proliferation, metabolism, angiogenesis, erythropoiesis, and so on. So these transcription factors has been studied widely, especially in the hematopoietic system, in the immune models. And here I'm just going to show you a brief overview of what is known. And here you can see the overexpression of HIF1 alpha, that is the main player of this pathway, is specifically in the more stem cells of the immune system, the LSK, CD34 negative cells. And when they conditionally delete in a genetic mutant model these this protein, they found in secondary transplantations that these cells failed to engraft, leading to concluding that this protein has a very an important effect in the maintenance of the capacity of engraft and the cyclone of these cells. But just to show you a little bit of the controversy that exists in the field, some few years later, the group of Camille Kang published this paper in which they used a similar model, and they failed to see this effect both in primary and secondary recipients of a similar murine model in none of the different differentiated or more stem populations of the hematopoietic system of these mice. But what happened in the hematological malignancies? So just here I show you a paper in which they delete the function of HIF-1 alpha by a chemical compound with shRNA against HIF-1 alpha or overexpressing VHL, that is the protein I showed you before, that promotes the degradation. And in the 3 cases, they observed that these cells have a defect in the proliferation and maintenance of the disease, leading to the conclusion that these proteins are somehow important in the evolution and cycling of the cells. So all of these lead us to conclude, okay, let's think if we can use this pathway to stimulate the cells and to make them cycle and in AML patients, and to try to sensitize the cells to the chemotherapy and avoid the relapse of the patients later on. But as you may know much better than me, AML is not a single disease. It's a very heterogeneous disease caused by many different genetic alterations. So we really didn't want to study AML as a whole, but we really wanted to focus in specific cytogenetic subgroups in order to discover possible effects over this pathway of the different cytogenetic aberrations and mutations. For example, it is described that IDH1 and 2 can have some influence in the PHD enzymes and can stabilize the pathway. So first of all, we take advantage of the publicly available databases with RNA-seq, bulk RNA-seq data, and in collaboration with a group of Eduardo Eiras, we selected different cytogenetic subgroups: translocation 821, MLL rearrange, inversion 16, FLT3-ITD, NPM1 mutations, and normal karyotype without FLT3-ITD and NPM1 mutations. That were the groups that we found more prevalent in these databases. And we studied the transcription, how was the transcription of a group of genes, these 119 genes that are targets of the HIF pathway that we call hypoxia pathway or hypoxia signature. Here I'm showing you the PCI analysis. First of all, I'm just going to show you the target results because the rest were quite similar. And here you can see that when we analyze the transcriptome, taking into account the expression of all the genes, all the patients from the different cytogenetic subgroups were clustering quite together, so they were quite homogeneous. But when we focus just on the hypoxia signature, we could observe that the inversion 16 samples were clustering separately from the rest of the groups. When we analyze the expression of HIF-1 alpha and HIF-2 alpha, also known as EPAS1, we observed that inversion 16 group was the one expressing higher levels of these genes, comparing especially to MLL-rearranged leukemias that were the ones expressing lower levels. Also, we focus on translocation 821, since it's also a core binding factor leukemia, also with high, similar to inversion 16, and also showing higher levels of HIF-1 alpha and HIF-2 alpha. And when we analyze by genes and enrichment analysis the expression of these 119 genes, and we compare expression in these different kinds of cytogenetic groups, we observe a significant increase over expression in inversion 16 compared to MLL. But we failed to find it in translocation 821. So we decided to focus our analysis in these 3 cytogenetic subgroups, so inversion 16 as a HIF-high AML, MLL rearranged as a HIF-low MLL, and translocation 821, since it is also a core binding factor leukemia similar to inversion 16, but behaving differently in respect of the activation of this pathway. So we collect different samples from patients, and we managed to get 4 samples from MLL patients, 4 from inversion 16, and 3 for translocation 821. And we also wanted to study the evolution of the disease, so we focused also in samples that we could have material from the diagnosis and the relapse of the patients. And also we wanted to discard some mutations in FLT3-ITD and NPM1 and IDH1 and IDH2, since I told you before, could have some kind of influence over the pathway. So how can we study the hypoxia pathway in the leukemic stem cells specifically, since these cells are mainly defined functionally, so the cells that you can transplant into the mice and give rise to disease. So just not to be based on one or two surface markers that is known that they are not bona fide markers, we decided to go for single-cell RNA sequencing to try to use the whole transcriptome of the cells to identify and study more in detail this pathway in the specific population of the stem cells. So basically, we took the different cells from the samples, and we sorted based on CD33 and CD45, the blast population, and we focus specifically in the CD34 positive, CD38 negative, which is described to be enriched in leukemic initiating cells in AML. We knew that just focusing on this specific population, we are missing or losing the analysis of the whole sample, the complexity of the whole sample, but we wanted to enrich the analysis in these cells because normally the percentage of them in the samples are really, really low. And as a control, we also studied the CD34 negative, CD38 positives to be the opposite population, more differentiated populations within the leukemia to have some kind of control. So we took the cells and we came here to the CNAG and we did the single-cell RNA sequencing with the 10x genomics technology and Illumina sequencing. And we did a posterior bioinformatic analysis to try to be able to identify the LSCs and to study the hypoxia pathway. And we took advantages of the LSC6 score, that is a score formed by 6 different genes described in this paper. And also we took advantage of the atlas from hematopoietic cells described by Van Galen et al. And Trian et al. That I'm going to show you later. So this is the UMAP from the cells that we sequenced. Here you can see the cells from the inversion 16 samples, the translocation A21, and the MLL. So we decided to merge together cells from the different cytogenetic subgroups, but separately, each of the group. And in red, you can see the cells that we were interested in, the CD34 positive, and in blue, the control population, the more differentiated, the CD38 positive. And we managed to sequence around 20,000 cells from each of the cytogenetic subgroups. So first of all, we checked the expression of CD34, so to check that the cells that we were sorting, they were effectively expressing CD34. And we, to identify which of these cells within this population were the LSCs, or to enrich a little bit more in this population which were the LSCs, we took advantage of this score that I told you before. And the way we did is we took each sample one by one, and we do a random clusterization, and we divide all the clusters according to the score of this signature. And we pick as LSCs, so the stem cells, only those ones over the median of these expressions. In this case, it will be cluster 9 and 13 that are here. Here I'm showing you that the cells with the higher scores, they are also falling into the same location as the CD34 positive cells. But to decide, in case of questions or doubts that we couldn't decide which of these clusters were the ones highly expressed in the LSCs, we took advantage of these 2 atlas that I told you before, that they did single-cell RNA sequencing in different hematopoietic cells, and they characterize different phenotypic status of the whole hematopoietic system. So in this way, we can identify different status, so HSC-like cells, because in this case, they are not healthy, they are leukemic, progenitor-like cells, and so on. And for example, in the sample I showed you before, among the cluster 9 and cluster 13, we decided to pick only cluster 9 because, as you can see here, this is the one enriched only in HSCs and progenitors, but the cluster 13 was enriched in more early erythroid cells. So this was one of the atlas, and this was the another one that it was quite similar, so you can see that the more stem cells were located in the same place. So with all this information, we decided which cells from each sample were enriched more in LSCs, and we divide all the cells in these 3 groups. So the cells identified as LSCs, we call it LSC34. The ones that we sorted as CD34 positive but not identified with the signature non-LSC CIS34, and the control cells, the 38 positive cells. Here in inversion 16, you can see an additional LSC cluster, which belonged to only one of the samples that we decided to analyze separately because they were falling differently. So we decided not to mix all the cells. And yeah, we were checking with different signatures if these were effectively, if we're really identifying where were the LSCs. Here I'm showing you a pseudotime, so you can see like a continuum of differentiation from the more stem to the more differentiated cells. And we also use other additional stem cell signatures, like the one described by Gentles et al. And Epper et al. And here you can see that the cells that we identify as LSCs were displaying higher levels of these signatures compared to the more differentiated cells. Also, we checked that to avoid a possible contamination with healthy cells from these patients, because the patients also has healthy cells in the bone marrow, we analyzed the expression of different genes that are known to be overexpressed in AML cells. And here I'm showing you these two, CLEF12A and JUN-D. And when we compare the expression in our cells with the healthy cells with data from the Human Cell Atlas, we observed that these genes were expressed comparing to the healthy cells, which makes us think that mostly of the cells that we analyze here are effectively AML cells and not residual healthy cells that can be contaminated in the analysis. So we decided to analyze some other metabolic properties just to be sure that we have LSCs. The first thing was to check the quiescence status, of course. We took advantage of these two data signatures in which they describe that you can identify G0 cells in G0 phase. And here you can see that our LSCs, identify LSCs, they display higher levels of these G0 signatures compared to the more differentiated cells. An additional metabolic pathway is important for the leukemic stem cells and the myeloid leukemia. I'm sure you know of this, but I just want to focus in the ROS pathway, which is, is described that especially leukemic stem cells are low ROS. And here you can see that in the 3 cytogenetic subgroups, we can detect low ROS levels in the more stem cells in the identified LSCs compared to the differentiated cells. I also wanted to highlight the ER stress pathway, which is described to be overexpressed in the LSC compartment, but we only could confirm this in the MLL cytogenetic subgroups, and seems to be the inversion 16 and translocation A21, they are behaving differently respect to this pathway. Yeah, well, also we checked which genes were differentially expressed within the LSC compartment in the different cytogenetic subgroups, and we found some like CD34 and FAM38 that are the ones included in the 6-gene signature. And now in the lab, currently we are trying to explore more the biology, the influence of these genes in the biology of the LSCs and the initiation of the leukemia. But this is a different project. But what happened with the hypoxia, that is the one that we're interested in us. So when we analyze the hypoxia pathway within our cells, contrary to what we were expecting, we observed low levels of the expression of HIF1 alpha and also the hypoxia signature, these 119 genes that we were analyzing previously. And here you can see the quantification of this expression of this pathway. So in our LSC cells, this signature was lower expressed compared to the more differentiated cells. So to discard a possible bias of having this big signature with this many genes, we also analyzed the expression of 5 additional signatures that is described in the literature that are overexpressed under hypoxia conditions. And here you can see that the tendency is quite similar to our hypoxia signature, with lower expression in the stem cells and much higher in the more differentiated cells. So we decided to analyze gene by gene because maybe it's not the whole pathway which is activated in the stem cells, but specific genes. And here you can see that this was quite heterogeneous in the different cytogenetic subgroups and in the different populations. So there are specific genes, like for example, MPN1 or CD99, that are consistently upregulated in the 3 cytogenetic subgroups in the LSCs compared to the differentiated cells. But there are others that are specific for each of the cytogenetic subgroups. But still, we analyze the expression of these cells in comparison with the healthy cells. And here, when we analyze total bone marrow cells or a specific stem compartment, we observe that this pathway is highly activated in our leukemic cells in comparison with the healthy cells, which still lead us to continue with exploration that this pathway can be exploited in therapy. But what happens also in relapse evolution? Because you remember I told you we pick also samples paired at the moment of diagnosis and relapse to study what happened in the evolution of the disease of these patients. So in a similar way, we managed to sequence around 20,000 cells from relapse material. And in this case, instead of plotting together the cells from the different cytogenetic subgroups, we decided to analyze patient by patient, so comparing the cells from the same patient at the moment of diagnosis and relapse. And here you can see one patient, another from the different subgroups. We identify in the similar way where the LSCs were located, at the relapse and the diagnosis. We identify with the LSC signature and the ALATs in the similar way as I described previously. And here you can see the evolution of the stemness score and the hypoxia score in the different pairs. So for example, in this pair, in the relapse, we observe an increase in the stemness score at relapse and a decrease in the hypoxia score. And to put all the data together, we generated this plots, and here you can see the 7 pairs. What we observed that it was not a general tendency or a general behavior of these pathways in the relapse of the patients. So in some of them were upregulated and in some downregulated. But what we observed is that these 2 pathways, so the stemness score and the hypoxia score, were always inversely behaving inversely. So for example, when the stemness score is upregulated in relapse, the hypoxia is downregulated. And vice versa, when the stemness score is downregulated, the hypoxia score is upregulated. This was happening in 6 out of the 7 samples that we analyzed. We also analyzed the different metabolic pathways, and you can see also that there is no a general tendency. But when we analyzed the genes that were overexpressed in relapse samples, and we did a gene ontology to see which were the pathways upregulated, we found in all the relapse samples a tendency to appear terms like translation initiation, elongation, ribosomal, and so on, which make us think that could be an interesting pathway to further explore in the future in these cells. Okay, so we failed to find an increased upregulation of the hypoxia in the stem cell compartment, but we found an increased expression respect to the healthy cells. So still we need to perform some functional assays to see if we can sensitize the cells inhibiting this pathway. So we use this compound, B87, that is reported to inhibit accumulation of both HIF1 alpha and HIF2 alpha. And we perform several in vitro and in vivo assays. So briefly, I'm going to show you some of the results. For example, here in vitro, we plated our primary AML cells over a layer of MSC5. Here you can see well, I don't know if you can see it properly, but here these bright cells are the leukemic cells that are floating in the plate, and here you can see some cobblestones, some cells, leukemic cells that go below the MSC5, forming these areas that indicates that the cells are like finding their needs, their proper place to grow and expand. So after several days in vitro and after the recovery of the cells after the thawing, we treat the cells in hypoxic conditions with the inhibitor or the vehicle and the cytarabine, that is a normal drug used in clinics, or the vehicle. And we perform different assays to assay the initial capacity after the treatment of the cells. I'm just going to show you the results from the long-term initiating cells, the long-term culture initiating cells assays. So here, after the treatment, we plated different doses of cells in different number of wells, and after 5 weeks, we score the wells for positive or negative wells. And with a software called LDA, we can estimate the amount of initiating cells at the beginning of the culture. So here you can see that the cells treated with a combination of RAC and the inhibitor, we found 1 out of 1,400 cells, and if we compare with the cells only treated with the RAC, we found 1 out of 300 cells. This is a reduction of 3 times in initiating cells when we treat with both compounds. This is a summary of the 6 samples that we performed this kind of analysis, and here you can see that we found a significant and consistent reduction in all the samples when we treat with the inhibitor in combination with ARC when we compare to only the cytarabine alone. And in vivo, we performed similar assays. So first of all, we transplanted the mice with the AML cells. And when the mice were engrafted with similar levels, we treat the mice with the RRC or the vehicle and the inhibitor of the vehicle. And we perform different assays to see the initiation of the cells. Here you can see that after the treatment, we can observe a significant reduction of the engraftment of the human cells in the mice treated with both compounds, the inhibitor and the cytarabine, respect to the other groups and specifically to the mice treated only with the cytarabine. When we pick the cells from the bone marrow and perform colonies to see the initiation capacity of the cells, we observe a twofold decrease in the number of colonies that we observe in the cells coming from the mice treated with both compounds. And what we observed is that these colonies were really, really much smaller than the rest of the conditions. So we counted the total cells of the plate, and we found a significant reduction of the number of cells in the place coming from the cells treated with both compounds. Also, we checked by FISH that effectively these cells and these colonies were coming from leukemic cells and not from murine cells or residual healthy cells from the patient. And finally, we performed secondary transplants in a similar way that we did the in vitro assays to quantify the frequency of initiating cells in the samples. And we transplanted different doses of cells in a different number of mice. And here we observe a significant reduction of the number of initiating cells in the samples coming from the mice treated with both compounds, especially compared to the mice treated only with cytarabine. And in longer time points, we observe a much higher reduction of the engraftment in these mice. So finally, just I want to summarize all the results. And we have basically generated a single cell atlas, an RNA sequencing atlas, from AML cells enriched for LSCs, comprising around 100,000 cells from these three cytogenetic subgroups. We have managed to study the hypoxia pathway, specifically in this compartment, but we found a lower status of this pathway in the more stem cells compared to the non-differentiated, but still this pathway is much highly activated compared to the healthy cells. And we managed to modify this pathway chemically to sensitize the cells in combination with the standard of care chemotherapy to efficiently eliminate these leukemic stem cells in the AML. And finally, I would like to thank all people in my group, Pablo's Menendez group, especially to Pablo and Juan Luis, who is the bioinformatician who has done all this bioinformatic analysis, and also collaborators, especially Holger, of course, and all the collaborating hospitals that has provided us with the AML samples to be able to do this study. So thank you very much for your attention. I don't know. Maybe we can ask the experts.
Yes, thank you, Talia, for this interesting. I was wondering about if you have any hint on the toxicity on normal hematopoietic stem cells, if it had just to bring the compounds to the clinic.
This is something we are now performing in the lab. So we are basically performing same experiments, but with cord blood cells, CD34 to see if the toxicity when we put the both compounds is increased that when you just put cytarabine. But this is something we are ongoing and we are performing now. Yes.
So that's what you're going to do before you do the clinical trial.
Exactly.
Other questions? Elias.
Thank you for the nice presentation. When we detected the phosphorylation oxidative pathway in the Richter transform cells that have been treated with many samples, looking in the literature, I came across the studies by Jean-Emmanuel Sarri in Toulouse that he has detected high phosphorylation oxidative in resistant AML cells, suggesting very interesting, that could be, to me, that could be a more general phenomenon that in hematopoietic and hematological cells. So I was wondering, I didn't see if you detect this pathway in in the LC cells, or you had also some cells in relapse that I guess was at the different treatments. Can come across this pathway in your samples, or?
So basically, this is RNA sequencing. Of course, it's RNA, so it's not protein level or functional level. But when we analyze different genes involved in oxidative phosphorylation, here you can see that, well, even the differences are significant because this is thousands of cells. We couldn't find dramatic differences among different compartments, so between the stem cells and the more differentiated cells. So in this analysis, if something is happening, is a little bit less activation in the stem cells compared to the more differentiated cells. But of course, this is at the level of RNA and not at the level of protein and functional. But yeah, this is something that we were also trying to look for. So what happening with the glycolysis and the phosphorylative oxidation, because these two pathways are really interesting in the field of the stem cells, and especially glycolysis because it's quite related to the hypoxia pathway as well. Yeah.
Thank you.
Okay, I hope you enjoyed talking to all of your colleagues. I did. So we will continue. We have now Jaime Font de Mora from Valencia, who's going to talk to us about liquid biopsies and so on. Okay.
Well, first of all, thank you very much, Ivo, for your kind invitation to come to the 11th symposium at CNAG, and allow me to share with you data that we have on genomics in neuroblastoma. Neuroblastoma is a pediatric type of cancer with around 80 new cases every year in Spain, 900 new cases in the European Union, and around 700 new cases every year in the US. It's a rare type of cancer. And neuroblastoma arises from cells of the neural crest, which is this temporal structure that forms during embryogenesis, while the closure of the neural tube, and the cells are going to migrate dorsoventrally to innervate and differentiate into other type of tissues, including calcitonin-producing cells in the thyroid, Schwann cells, melanocytes, primary sensory neurons, and importantly for neuroblastoma, neuroblasts that are going to generate the sympathetic system. This is the clinical staging for neuroblastoma that was generated over a decade ago by an international consensus group. There are stages for L1, L2, M as for metastasis and MS. These clinical stages, together with age at diagnosis, 1 year and a half, less than 1 year and a half is of better prognosis, and together with the histological category and plus the differentiation, this is also very important because actually the differentiation of the cells, you can distinguish 3 different groups, neuroblastic groups in this category. You have those that are small rounded blue stain cells that form rosettes, very poorly differentiated, by the way, that are characteristic of neuroblastoma. Then you have a higher degree of differentiation, ganglioneuroblastoma, and even more differentiated cells, the ganglioneuroma, and of course, of better prognosis. And for the first time, it was introduced genetic alterations. Miken amplification is by itself of very poor prognosis, and 11q deletion and diploidy. And all these variables, they define the risk in neuroblastoma. The risk means the risk to relapse, the risk to overall survival. We are specifically interested in the high risk. We want to solve problems. And in the high risk, what we have is MICEN amplification. Some of them have MICEN amplification. You can neatly see in these SNP arrays as a very sharp, well-defined magnification in 2P, where MIC is located. And also you have, well, it's mandatory for the diagnosis to perform a FISH analysis. And then you have those others that have 11q deletion, and also very well detected by SNP arrays with different size of deleted in 11q, different size of region. And it is interesting that usually MICEN amplification do never occur or barely never occur with 11q deletion. Either the patient has MICEN amplification or has 11q deletion. And this suggests that perhaps they are both independent drivers for the disease. Still, what's in 11q that is making the disease is not known. We have in the past been interested in analyzing the Spanish cohort and observed that 11q deleted neuroblastomas, those in blue, in panel A, are subjected to permanent relapses, constant, even more relapses than MICEN amplification in red. And although the overall survival, now panel B, is not as severe as those with MICEN amplification, it is still of very poor survival. So something has to be done. In the latest years, several pangenomic analyses have been performed. And it is noted that high-risk neuroblastomas, those cases in gray in this upper row, the high-risk neuroblastomas, they have higher number of somatic, exonic, non-synonymous mutations than those in the lower risks groups. But still, there are very few point mutations. There are from very few, like 3, 4, 5, up to the average is 20, 23, up to 50 mutations in the whole genome. So, and still that is in the medium among the pediatric tumors. Neuroblastoma has lower mutations than high-grade gliomas or osteosarcomas, and even way much lower mutations than adult tumors. So when you analyze for driver mutations, yeah, it is known that MICEN amplification is a driver mutation in neuroblastoma. ALK amplification, ALK, it's also a driver. ALK point mutations have also been defined in neuroblastoma, and there are other few genes involved in the activation of the RAS pathway. But there are cases in which you don't find drivers. So our starting hypothesis was that perhaps there are epigenetic changes, mostly DNA methylation, that are relevant for the etiology of high-risk neuroblastomas. And then our second thought was that liquid biopsy, and by that I mean DNA in the plasma of the blood that contains information for prognosis, or as well biomarkers for the better stratification of the disease, and finally, it's a beautiful technique because it's non-invasive and you can monitor the patients very easily. So this is our strategy. We got patients at diagnosis, 1 to 2 milliliters of blood. More than that is not ethically allowed. And from the blood, the plasma, from the plasma, the nucleosomal DNA, and that DNA was then analyzed for quality controls. And by that, I mean quantity and integrity of that DNA. And at this point, I have to say that 1/3 of the samples did not commit our quality controls. We have to discharge them because the quantity was very low, or perhaps even it contained high chromosomal DNA degradation smears that we couldn't use it. So those that did commit our quality controls, then they were subjected to enzymatic conversion of unmethylated cysteines, followed by whole genome sequencing, and then data mining and analysis. And this is our cohort. We named it Liquid Hope, as for the European project in which we were involved, or we are still involved. The cohort contains 64 patients at diagnosis, 22 of them with MECAN amplification, 34 with 11q deletion, and 8 with other alterations. And we first wanted to ensure that our samples were actually tumor DNA. So we had to internally validate our samples. And secondly, once we generated data, we wanted to validate our samples with external cohorts, target and Westermann cohorts. So we initially started with doing a principal component analysis, a PCA analysis, and noted that there were 9 samples in yellow that clustered together with the non-tumor DNA controls in green. This suggested that perhaps these samples did not contain tumor DNA. And actually, when we came to the analysis for the copy number variations, these samples didn't show any copy number variations at all. So we had to conclude that probably there was some malpractice during the procedures, the handling of the samples, and we had to discharge also these samples. But we did observe that the other samples, we could validate the initial diagnosis. Those that had MECAN amplification, we could confirm that actually there was MECAN amplification in our analysis of the blood samples. And those that were diagnosed with 11q deletion, yes, we could see it neatly. And importantly, those with MECAN amplification in red, they split apart from those with 11q deletion in blue. This supports that there are specific CPGs that differentiate between both subtypes. And first, we wanted to see whether our way of analysis, which is whole genome sequencing with enzymatic conversion of unmethylated cysteines, were comparable with the other cohorts that were done with the chemical conversion, the bisulfite conversion, and arrays. And this is what we got. We did the average methyl cysteine for each CPG in our cohort, the Liquid Hope cohort, and compare it with the average methylation level in the target cohort. And they both, or they are aligning, they are making a pattern, a line pattern, confirming that they are miscible. We can compare both cohorts. So we went back to our cohort and developed an elastic net penalized regression model to identify those samples that had MECAN amplification in red and those samples that had 11q deletion in blue. And the model predicted beautifully, perfectly, of course, perfectly, the validation in our own cohort. But importantly, it also predicted with high accuracy the MECAN amplification and 11q deletion in the American cohort target, as well as in the German cohort Westermann. These results, what are telling us is that liquid biopsy is a very feasible technique for epigenetic studies, and that there are a signature of 52 CPGs that distinguishes between both subtypes. And probably those CPGs, those methylated CPGs, are relevant for the etiology, for we cannot say driving, but they are accompanying because they are differentially methylated, they are accompanying each tumor subtype. Among those 52 CPGs, 8 had as well prognostic value. And among those 8, there were 2 that in addition were located in 2 druggable targets. One is CCR7 in the intron 1 of CCR7, and one in the promoter region of the colony-stimulating factor 1 receptor. They are both hypermethylated, the blue boxes, hypermethylated in 11q deleted neuroblastomas, no matter which cohort you were looking at. And the hypermethylation meant worse prognosis. So because CCR7 is highly overexpressed in naive T cells as well as in memory resting T cells, and CSF1R is highly expressed in monocytes and importantly in macrophages, we then wanted to do the convolution studies in order to identify the subsets of immune populations that are within each tumor. And I have to apologize because now I have changed the colors. Now the 11q is in red, MIC-10 is in blue, and green others. And what we noted is that 11q deleted the neuroblastomas are surrounded or contain higher number of resting memory T cells. They have also increased number in regulatory T cells, that is immunosuppressant cells, and also they have higher number of all subtypes of macrophages, from M0, M1, M2, but more significantly in polarized M2. So we then moved to perform co-cultures. What we did is we used patient-derived organoids, and we co-cultured them with unpolarized M0 macrophages, and noted that 11q deleted neuroblastomas had very potent capacity to polarize macrophages to M2. So we came up with this model in which 11q deleted neuroblastomas, they have a higher enriched immunosuppressant microenvironment. Why? Because they are overexpressing, I didn't show you, but they overexpress high levels of PD-L1 and higher levels of indoleamine deoxygenase 1, IDO1, TGF-beta, as well as interleukin-10, that are driving the differentiation of naive T cells into regulatory T cells, immunosuppressant cells, and polarizing the macrophages into M2. And perhaps, we hypothesize that perhaps immune modulators targeting CSF1R and CCR7 are going to be very important to maintain the undifferentiated or unpolarized populations, and will be beneficial in the immunotherapy that is currently being given to the high-risk neuroblastomas. But now going back to the genomic data, what we observed, or we studied the differentially expressed genes in 11q deleted neuroblastomas by their chromosomal location, and observed that 11q deleted or 11q has the highest number of differentially expressed genes. This is something that, of course, it was expected. Yes, they have the highest number of differentiated genes are located in 11q. But in addition, in 17q, and this is also expected because 17q gain is very frequently associated with 11q deletion. And importantly, there are 4 genes that are relevant for the subsequent studies. There are 4 genes that are repressed or with lower expression levels in blue in the 11q deleted neuroblastomas in comparison with MIC-10 amplified neuroblastomas. And those genes are ATM, H2AX, CHEK1, and MRE11A. Those 4 genes are located in 11q, and they all belong to the DNA double-strand break repair machinery. The differential expression of these genes between 11q and MYC are not due because the promoter is hypermethylated or methylated at all. Actually, we didn't compare in different cohorts. It's always the same. So we must conclude that there may be other epigenetic reasons for this downregulation of expression, or simply because a gene dosage effect. And we perform, in order to identify genes relevant for 11q deleted neuroblastomas, we combine double studies with panel sequencing together with SNP arrays to define which was the minimal shared deleted region among different 11q deleted neuroblastomas. And we came up with the observation that always in that minimal deleted region, it was included ATM, H2AX, and CHEK1. And when we analyzed the other chromosomal sites, we observed that 11q deleted neuroblastomas had the highest number of segmental chromosomal alterations within the high-risk group, more than MYC or more than others, supporting the idea that, yes, there are defects in the DNA repair machinery in 11q. So we went to use cell lines and observed that 11q deleted cell lines, those in the middle and in the bottom, they have lower expression levels of ATM, and they were sensible to PARP inhibitors. We had to use DNA damaging agents. We used temozolomide, which is an alkylating agent for the DNA. And those are the blue, or the purple lines. Not always occur that. We demonstrated that these cell lines are sensible to PARP inhibitors plus DNA damaging agents only if P53 was integrated. So if it was not, they were not sensible to it. And this also could be nicely reproduced in xenografts in vivo, in animal models. They are highly sensible to the concomitant treatment of PARP inhibitors and temozolomide. So we end up concluding that 11q deleted neuroblastomas are sensible or are subjected to, or they could be treated with PARP inhibitors plus DNA damaging agents only if they don't have P53 mutation. And since at diagnosis, barely no neuroblastomas have P53 mutation, what we think is this therapy would be beneficial if it is as a first line of treatment. Now, we wanted to generate an animal model for the disease. And for that, we had to integrate in cis, that is in the same murine chromosome, ATM, H2AX, CHEK1, all of them driven by the tyrosine hydroxylase promoter CRE that will allow the specific deletion of the genes in the neural crest. But based on the more recent knowledge by Matthias Fischer's group that supports that the higher risk is due to the TERT activation or telomere lengthening. It could be either by TERT activation, different ways of TERT activation, or by alternative lengthening of telomeres out. So what we did, we also included the overexpression of TERT in the neural crest. And these animals, they still have normal development of the medulla of the adrenal gland. The medulla of the adrenal gland comes or arises from the neural crest. Now, this is a crest staining to show where it's located. normal staining. There is no hyperplasia, nothing. And, but what we did note is that these animals, they have poorer or defects in the innervation of the sympathetic system in the distal colon. Moreover, in the process of generating these animals, we also had to generate double knockouts for ATM and H2AX. And those animals are smaller, 30% smaller in size, and the size, or this reduction in size, in the dark blue lines, they persist through adulthood. So some of these animals with lower, I have to say that it's with lower penetrance, but some of these animals do develop Hirschsprung disease, which is, it's a disease due to the lack of innervation of the guts by the sympathetic system. So actually we are getting very close, but something is missing in this picture. Something is missing. We believe it's inflammation. Inflammation is important for almost most cancers. Inflammation may come from outside sources, such as exposure to heavy metals, or bisphenol A, or infections, and as well for internal sources, such as body mass index or gestational diabetes, etc. So our current working hypothesis is that 11q deleted neuroblastomas are a multigenic disease containing specific epigenetic and genetic alterations that recreate a niche, an immunosuppressant niche that enables the resistance to the current therapy given to high-risk neuroblastomas. And we also think that the onset of the disease requires 3 different branches. The first one is the DNA repair defect. It's a combined haploinsufficiency of poor expressed genes involved in the DNA repair. The second one is due to stemness. In our case, the overexpression of TERT, and it's very frequently observed, the TERT alteration by SNP arrays or translocations or overexpression in neuroblastomas, in the high-risk group. And the third one is inflammation. Our proposal is to do endogenous inflammation. How? With the matioficin 2 mutation or deletion that we borrowed from Antonio Zorzano, and that will generate, and now in the 2 previous talks, endoplasmic reticulum stress and ROS, reactive oxygen species production, has been spoken in 2 other diseases. And here as well, we believe that this endogenous inflammation will be the trigger for the disease. So as for conclusions, I have shown you that 11q deleted neuroblastomas are a unique genetic and epigenetic entity distinct from any other. They arise from different sources. That combined haploinsufficiency is a hallmark for high-risk 11q deleted neuroblastomas that could be treated differently as current treatments, for instance, with PARP inhibitors and DNA-damaging agents, or at least combining current immunotherapy with immune modulators for the colony-stimulating factor 1 receptor or CCR7. And our model provides support for using liquid biopsy as an optimal strategy to investigate, to further investigate methylomes, but also to implement precision medicine in the patients. Now, for acknowledgements, the people in the lab, mostly to Eva Maria Trinidad and Esther Coronado and Quique Vidal, who is not here, and our wonderful CNAG collaborators, Simon Heath and Anne Esteve in the data mining, and Marta Good in the sequencing, and the support, the financing support, mostly but not only, It was supported by a European grant, Transcan, and the Fundación Científica AECC. Thank you very much.
Thanks very much. Very nice. Very nice. Questions?
Hi. Yeah, very, very interesting. So I have a question out of ignorance of the treatment of 11q deleted neuroblastoma. Are they treated with radiotherapy? Because radiotherapy is known to also induce double-strand breaks, and this could be particularly sensitive, right? Because considering that it has this deletions of 11q.
I should have brought, I'm not a clinician, and I don't think they know. I would say no. It's not, radiotherapy is not involved in it. There is a chemotherapy induction. Kojek is named, I don't know if the different chemicals in Kojek. And then there is the ablation of the medulla, and then there is the immunotherapy with anti-GD2 antibodies. The American group, the Children's Oncology group, is using together with the immunotherapy, with the anti-GD2, so it's a ganglioside in the surface of the neuroblastoma cells. They are using, together with it, a granulocyte macrophage colony stimulating factor, which has a potent capacity to, or they have shown it is very robust in the treatment. And they also use, they intercalate interleukin-2. In Europe, when it was imposed a few years ago, the antibody therapy, it was not included those because I am not sure, but I think it was not available for Europe, the granulocyte macrophage colony stimulating factor. And that is the therapy. I don't recall at all that there is radiotherapy. I don't think so.
Okay. No, it was just curiosity. Well, thank you.
Thanks a lot for the talk. That's a very nice talk. I have two questions. The first one is related to if there's like these two types of amplification and 11q, if phenotypically, if there is any difference or that they're not much distinguishable, if it's like something that's only genetic or it can be related to something more like, phenotypically on the patient. And the other question is about the origin of these neuroblastomas, that you mentioned that there could be something related to inflammation. And if there's something besides like in the developmental stages that create these mutations, like these MECAN amplifications and so on, there could be something on the pregnancy that could actually sort of end up of like having neuroblastoma. And the question here specifically is, have you observed mutations in normal tissues on these like MECAN amplifications in 11q? If there has to be a combination of both genetic and environmental, or if you have any thoughts on that.
Wow, very interesting question. For the first question, no, they are not distinguishable if you have, or morphologically, you cannot distinguish if they have MECAN amplification or 11q deletion. That's one of the reasons why you have to do, it's mandatory, the FISH analysis by MECAN. Actually, sometimes MECAN can be amplified in foci, like in islets. And it is recommended that the patient is being treated as MECAN amplified, but still there are doubts in groups. So no, there are no morphological changes. And MECAN amplification, it's heterogeneous sometimes. And as for the second question, I remember when I was a postdoc at Dana-Farber, and I heard one wonderful talk, how if you modify the stroma cells, then cancer cells would not develop. I cannot answer your question. I think it is relevant. Maybe there are mutations, modifications, or epigenetic modifications in the stroma that are allowing neuroblastoma to proceed and even to protect from the therapies, but I don't know. Nothing is known about the stroma cells in neuroblastoma.
Thank you very much. Thanks a lot.
Thank you.
Núria López-Bigas, long-term collaborator here. Also someone from 12 years ago, 12 years total.
Okay, so thanks a lot for the invitation and for the whole symposium. Wow, I'm showing the last slide actually. Okay. Okay, so I'll talk about analysis of cancer genomes. And starting with this, that everybody here knows that if we sequence a tumor, we find a lot of mutations. If we are able to sequence the whole genome, we find on the order of thousands of mutations, depending on the tissue, a bit more or a bit less, but it's in any case very big numbers. And more recently, some groups have started sequencing normal tissue, and we realized that the numbers of mutations that you find in normal cells, not developing any cancer or any tumor, it's also on the order of thousands. So one of the big questions in cancer research for the last 30 years or so is to find which genes and which mutations are the ones that drive tumorigenesis. At the end, cancer is a genetic disease caused by mutations, so which ones are these mutations? And one thing that we could do is, of course, going to the literature, because there are accumulated evidences for many cancer genes for many years now. And we know that TP53, BRCA1, MYC, APC, and many other genes are important. But in general, we have a scattered view. Like there are some genes that we have studied very well, some other genes that people have paid very little attention. We know that some genes are important in one cancer type, but we don't know what happens in the others. So what we thought is, if we could try to gather this knowledge of cancer genes across cancer types in a more systematic and unbiased way. And for us, this means going directly to the somatic mutations in thousands of tumors and learning directly from this data, not from the literature, but from the somatic mutations directly. You know that there are a lot of projects that have generated this type of data. For example, International Cancer Genome Consortium, that Ivo was an important part, the Cancer Genome Atlas, Harwick Medical Foundations, and others. Basically, they get a sample, a tumor sample, and another sample from the same patient. Most of the times, this is blood. And then they sequence and identify the somatic mutations, either whole genome or whole exome. What we have done is to gather this data from the literature, that data that is available. And for the analysis I'm going to show you today, we analyzed 28,000 tumors from different cohorts on different institutes and different cancer types. And we want to use this data to identify cancer genes. And the way is thinking that at the end this is an evolutionary process characterized by variation and selection. Then we can try to identify signals of positive selection in the pattern of mutations. So this is always a cohort analysis where we start, for example, with 300 breast cancers. And we ask, does this gene has driver mutations? So it's a cancer gene. And then we ask if it has more mutations than expected. Right? For example, if we look at TP53, there will be many samples with TP53 mutations, but the expected based on neutral mutagenesis is probably going to be very low. So we have many more mutations than expected. Or if we see that mutations cluster in particular hotspots or regions, that's another signal of positive selection. Or identifying that there is a bias towards high functional impact mutations. For example, that many mutations truncate the protein, stop codons, or the most important and conserve amino acids. So this is the concept of all this bioinformatic analysis that from a list of mutations in a cohort of patients can highlight which genes have positive selection. Some methods, these are the methods that we use: OncoDrive FML, OncoDrive Clustel, DNDS, CBase, mood panning, hot maps, and SM regions. Some developed in our lab, but some others actually developed by other people here in the audience, from mood panning and CBase, or DNDS from the Sanger. And what we realize is that combining these different signals of positive selection, it's the most comprehensive way for us to identify cancer genes and the more reliable way. Okay, so now we have a pipeline that goes from the somatic mutations in a cohort of tumors, runs 7 methods, combine the results, and end up with a list of genes. And we do this for all these cohorts. There are 221 different cohorts that this contains these 28,000 samples. And this way we are generating a compendium of cancer genes across cancer types. At the moment we have 568 cancer genes in the compendium. The most important thing is that this is a list of cancer genes for each tissue. So we have a list for breast cancer, another for colon cancer, pancreas cancer. One good thing of this approach is that it recovers most of the well-known cancer genes, because if you check TP53, HRAS, BRAF, they have these signals of positive selection, but of course, because it is unbiased, it also has the opportunity to identify new genes, and also to link genes that we know that were involved in one tumor type, now seeing which other cancer types they are also important. This is work by many people in the group. Many of them, probably you know them. I see Ines here, and some people from my lab, Fran as well. So, you know, we have to develop methods, combine the results, prepare the pipeline, collect data. So that's why it's a team effort for many years. So I explained the concept of identifying positive selection kind of very quickly, but it's not that easy. And many people know here that the main difficulty is to compare the expected. I say that we usually say, what is the number of mutations that we observed, and what is the number of mutations that we expect? Or what is the distribution of mutations that we observe, and what is the distribution of mutations that we expect? Computing the expected, it's the most challenging part, because this is kind of simulating neutral mutagenesis, how the mutations occur without any selection. And that's not easy for several things. First, because different regions of the genome have different rates of mutations. So we find some regions of the genomes with a high number of mutations, some regions of the genome with low number of mutations. And this changes per tissue and per mutational process. So that's one of the reasons why computing this is not easy, and many of the developments on these methods is to get this the most accurate possible. The other thing that we have to take into account is the different mutational signatures, so that different mutation types and in different mutation contexts have different probability. So many of you may be familiar with this type of mutational signature plot, but basically what it shows, this is a probability, these bars, or a frequency, of mutation type, C to T in this case, in particular context. So taking into account the nucleotide before and after. And this is, for example, the UV light mutational signature. So if there's UV light creating mutations in cells, most of them are C2Ts in dipyrimidines. Okay? So this, you can now imagine, then when we simulate neutral mutagenesis in skin, this context will have a higher probability than other contexts. Okay? So this is how we have to simulate this neutral mutagenesis in taking into account these two things. But that's not enough, and some of the things that we have learned in the last years is that we need to understand better this, the way that mutations accumulate in the genome, and that's why we have dedicated some time to that. So in general, this depends on the way that DNA damage, like DNA damaging agents create lesions in the DNA or mismatches, and also the way that they are repaired. Right? If these lesions are repaired, they don't lead to mutations. If they are left unrepaired, in the next replication cycle can lead to mutations. And this depends a lot on chromatin conformation. And a lot of the efforts on this field is to understand why different regions have more or less mutations, and some of our contributions here is not only on the large scale variation, but also on the local scale variation. For example, we realized several years ago, that's work by Savari in the lab, now he is a PI in India, in Bangalore, that there is a high rate of mutations in melanoma in transcription factor binding sites. So when you have a protein binding to the DNA, there are a lot of mutations accumulating from UV light, like 5-fold increase of mutations compared to the flanking regions. And we also were able to learn that this is because impaired nucleotide excision repair when you have a protein binding to the DNA. Okay? So then you have an accumulation of mutations, but it's not positive selection. In a particular region, it's because of the mutational process accumulates many more mutations here by chance. We also were able to see that there is a periodicity of mutations in nucleosomes and linkers. So when we have a nucleosome, DNA wrapping around nucleosomes, you have higher rate of mutations, and in linkers, lower rate. Work by Joan Frigola showed that there is a lower rate of mutations, mostly due to mismatches in exons compared to flanking introns. For example, you see a plot like this, especially in polymutants, that Marta has also talked about the polymutant tumors. They have a high rate of mutations. Most of them are mismatches that are not repaired, and you see this decrease of mutations in the exons. In this case, we think that this is related to a higher activity of mismatch repair in exons compared to flanking introns. And work by Uriolpic show that there is a periodicity of mutations in DNA wrapping around nucleosomes. So every 10.3 base pairs, you have a higher, lower rate of mutations. This is in very well-positioned nucleosome regions. Okay, so if you want to read about all this, we wrote a review a couple of years ago, or 3 years ago already, that summarized a bit all that. Another thing that it's important, and we also have work, is that when we are analyzing tumors, some of the patients have already been treated, and then maybe you have a metastasis. So there is another process of mutation accumulations in some of these cases that is due to the chemotherapy itself. Some of the chemotherapies are DNA damaging agents, and they create lesions in the DNA, and cells that survive and don't die and replicate, they can create, they accumulate mutations. So we analyze metastases of treated patients, whole genomes, to identify some mutational signatures of chemotherapies. I'll just show you a couple here, capecitabine, this T2G pattern, and this is cisplatin that it was already known before, and we could identify a very similar pattern in metastases in human samples. So again, work by Uriol with Ferran and Annabelle in the lab. So we are still working on this topic, because for us, if we want to identify drivers, it's very important to understand every day a bit better how the mutations occur without positive selection. And Claudia, who is another PhD student in the lab, is now doing analysis of hotspots across tumors using whole genomes in outside coding regions. And we are trying to understand why some mutational processes actually create hotspots without being positive selected. Okay, we are trying to understand this a bit better. So this is the question. If we see a hotspot in a promoter or in a long non-coding RNA, we may tend to think that this is a driver because of many mutations accumulating there across patients. But we also have to think that there may not be a driver and maybe a more vulnerable site that just more likely to or more prone to mutate. So this is what we are trying to understand. I will not explain. We have very interesting results here, but I don't have time to talk about everything. So that's work in progress that at some point we will explain in more detail. Okay, so we need to understand neutral mutagenesis and how mutations accumulate to go back to our original question, that is, we want to identify cancer genes and driver mutations. Okay, I think we have a compendium of cancer genes that, of course, is not perfect, is not complete, but for the cancer types where we have more samples, I think that we have a good understanding of what are the genes, at least the most frequently mutated genes that drive tumorigenesis. But then we have another problem, that even if it's TP53 or EGFR or any of these genes that we know very well that when mutated drive cancer, if we find a mutation in a patient, that may be a driver or a passenger mutation. So TP53 also have passenger mutations. So this is another question, how to find the specific driver mutations in these cancer genes. Usually when we are more in a precision medicine scenario, in the clinic, we find a mutation in a patient in one of these cancer genes, and then you want to know if this is an important mutation. You may go to ClinVar or OncoKB, that these are the most popular databases to annotate these mutations, and you realize that most of the mutations are not annotated. So they are variants of uncertain significance. Most of the mutations that you find in cancer genes in tumors. They are not annotated, so you end up with, okay, this is a variant of unknown significance, I don't know what to say. So we thought that we wanted to contribute into this question on a similar mindset, like can we learn directly from all the somatic mutations that we have in many tumors to annotate which are the important mutations in each cancer gene. To answer this question of which mutations are important in a cancer gene, for example, in TP53 or in EGFR, you could do several things. One thing that could be done is experiments. So we could go and create this mutation, introduce it in cells, and see if these cells now have tumorigenic capacity, or they proliferate more, or they have some tumorigenic properties. You could even put these cells with the mutation in mouse and see if they develop tumors. That's a way to validate their tumorigenic potential. And you could even do that with deep mutagenesis. So you generate all possible mutations in a gene, and this has been done for TP53, for some RAS genes, and then do this in CRISPR libraries and test them all. This is very useful, but also, as you can imagine, it's very time-consuming and it's a big effort. And in any case, it's also an experimental system. So you are testing if this mutation included in a mouse can develop a mouse tumor, right? Our question is, if a cell in a human tissue can develop, with this mutation, can develop a human tumor. The best experiment would be to introduce this mutation in a human tissue and see if the tumor is developed. That's not something that we can do or we want to do, but this has already been done. So all the tumors that we have are exactly this natural experiment of mutations occurring naturally in normal tissues, developing tumor, and now we have the mutations and we know them. So using this and thinking of this as a natural experiment, we thought if we could exploit this data using machine learning approaches to learn which are the features of driver mutations compared to passenger mutations. So we are going to use the same data set, 28,000 tumors across 66 cancer types, the same one that I presented before. And this is work by Ferran Muiños, who is here. He is a brilliant mathematician in our lab and key person for this project. Fran, who was a postdoc in the lab, and he is also here. He is now in Utrecht. Well, now he's here, but he's working there. Abel and myself were the co-leaders of this project, and Oriol Pic, that now is in London. He was a PhD student in the lab. This is published. I'm not going to explain in detail how we do it, but the main concept is we created a machine learning model per cancer gene. So we don't want, or we didn't aim. At the beginning, we were thinking about this, and then we realized, okay, that doesn't make sense to create one model that fits all. Every gene and every cancer protein works differently. The features that define driver mutations are different for EGFR and for another gene, so we are developing gene-specific models. And we are also developing tissue-specific models as much as possible, because we realize that in some cases, for example, EGFR is one of the cases that we highlight in the paper, we find completely different models for glioblastoma and lung adenocarcinoma. So as much as possible, if we have enough data, we develop gene tissue-specific models. And for each one, we end up with a model that is able to distinguish between, to classify any mutation in the gene to say if it's a driver or a passenger, based on the features of the mutations. So in total, we have 185 models. And then we can do in silico saturation mutagenesis, which means that once we have a model for TP53, this is a model for TP53 colorectal cancer, we can generate all mutations in the gene. So starting from the first nucleotide, an A, we can mutate to a T, a C, or a G, and we ask for each of these mutations to the model, is this a driver or not? And we do it for all, so that at the end we have a map of where are the drivers. Okay, so I explained kind of very briefly. There are many more interesting things in the paper. But back to the practical question, if you are in a precision or personalized medicine scenario, you have a patient with a mutation and you have to decide if it's a driver or a passenger, and then you are in this case that many of them are not annotated. Now, if you use BoostDM, there are a good number of them that we can give an annotation if we think that this is a likely driver mutation in those genes. You realize that we still cannot fill up all the mutations, and that's because we don't have enough data to create models for all the genes, only for the most frequently mutated genes. But we hope that in the future we will have more data. So this is useful for the cancer genome interpretation. We developed in the lab several years ago a tool called Cancer Genome Interpreter that you input a list of mutations in a tumor, and then you have annotations of which of these mutations are drivers, and for that we use Intogen and BoostDM in addition to other things, and which of these mutations are biomarkers of drug response. For this we use expert-curated databases. Cancer Genome Interpreter is a tool that we develop in the lab in a research setting, and it's actually being used quite a lot in some clinical settings. But now we are moving into a more clinical implementation of this, and we have a new project that is starting in November. This is a EU project for 5 years that is called CGI Clinics. And this is for clinical interpretation, so further development and implementation of this cancer genome interpretation. These are the partners. There are clinical partners in Spain, in Germany, in France, and in England. These clinical partners are going to help us to implement it and further develop it to fit all the needs in the clinic for the interpretation. As technical development is IRB, ourselves, and also CNAG, principally for the data sharing step. And we also have patient associations that are part of the project because we also aim to reach the patients. I will explain you now what we mean by that. And Alira Health, that is a company that is going to help us in the regulatory process because we plan to end with a tool that is approved for clinical use. We identified 3 main problems and solutions that we want to add into this project. The first is that right now this interpretation, so you have a list of mutations, you need to make an interpretation and make a decision for the patient. This is done differently in every hospital. In some places they have a lot of experts, a lot of resources, and they do it more completely or more comprehensively, and in other smaller hospitals they may not have the experts, and then this is, so it's non-systematic and it's non-equitable. What we want to do is automatize the process and make it the most complete and fitted with the clinical needs, so that the CGI clinics will generate a report the most complete as possible, so that everywhere we will have a similar interpretation. Even if we put all the important databases and we do this very well, as we mentioned before, there are many variants of uncertain significance. There are not in any database, even if we put all the important databases. So for us, the way to go here is this automatic learning. So similar as Intogen and BoostDM, keep learning from the data. The limitation at the moment is, as I said for BoostDM, we cannot build models for all the genes. So if we gather more data, we can learn more models and more sophisticated models that could take into account context. So part of the project also aims, and that's why the role of CNAG is very important, is to share the information. So at the same time that you are including your mutations for cancer genome interpretation, that you are one click away to share this data for research. So that we can keep gathering data effectively to improve the automatic learning. That's kind of a very important goal. We'll see. The other is that the patients are usually unengaged in the sense that they are passive actors. So the project also aims to reach the patient, mostly for these two objectives. That's first that they understand a bit better what all this means, what these mutations mean, and also that they are aware that their data has value. The data, the mutations in their tumor has value, and that they can share it and they can participate in the research process by improving the interpretation of the next patient. That's why the patient associations are a very important part of the project as well. So as a summary of this part, now I'll check if I have enough time, told you about Intogen, BoostDM, cancer genome interpreter, and the evolution towards CGI clinics. I'm not aware of the time, but you may tell me if I have almost finished? Or I have to, okay. Feel free to stop me because... Okay, so in normal tissue, so we have been talking about cancer. Cancer is a clonal expansion to a tumor and a diagnosis of a cancer patients, but there are clonal expansions in our normal tissues as well. And one that many of you may know is clonal hematopoiesis. So this is a clonal expansion of hematopoietic stem cells so that we have clones in our blood. And this is a very similar process, right? There are somatic mutations in hematopoietic stem cells. Some of those drive clonal expansion. For example, DNM3CA mutations, TET2 mutations. So you have these clones. And these clones, this clonal hematopoiesis is linked to an increased risk of AML, for example, acute myeloid leukemia, but also cardiovascular problems and all-type mortality. Okay, so we thought that with our expertise, we could tackle the problem or the identification of clonal hematopoiesis genes and mutations in a similar way as we have been doing for cancer. So that could be like, can we generate a compendium of clonal hematopoiesis genes? Can we identify in these genes with BoostDM which are the specific driver mutations? And at some point, we want to reach into interpretation for the clinic for clonal hematopoiesis mutations. So that's not done. That's work in progress, but I'll tell you where we are right now. So to start, we need somatic mutations in blood, not in tumors, but in blood. But we can exploit the data that we have of cancer genomes to identify somatic mutations in blood, because as I told you, in all these projects, we have a tumor and normal sample. The normal sample, in most of the cases, is blood, just because it's the easiest to get. And usually what we do is to identify tumor mutations using blood sequencing as germline control. And now we could do the other way around. So to identify somatic mutations in blood using the tumor as germline control. So this is what we call reverse calling. And we have done it for all TCGA and Hardwick. So that for 12,000 samples, we have a list of somatic mutations that we identify in blood samples. And we can distinguish them from germline variants because we have a second sample that is the tumor. Once we have these somatic mutations, now we can use the same mindset and identify signals of positive selection in the pattern of mutations. And we actually used the same pipeline, IntoGen pipeline, and identified 64 clonal hematopoiesis genes. These include all the clonal hematopoiesis known genes, like TET2, DNM3CA, they all have these patterns. And the results are already available in intogen.org/ch. And the paper is in bioRxiv, and it has now been accepted in Nature Communications. So that's work by Abel and Uriol. Second question, now we have a list of genes that we can keep completing it. We don't think that is complete at all. But now we have the second question, right? Once we have DNM3CA, for example, we know that mutations in DNM3CA drive clonal hematopoiesis. Which mutations in DNM3CA drive clonal hematopoiesis? So what Santi De Maggio, with the help of Ferran and Abel, are doing is using BoostDM to create models for each of these genes. So for the moment, we have enough data for 12 clonal hematopoiesis genes, and we have already generated some models for that. This is the case of DNM3CA, and then we can do the in silico saturation mutagenesis. So create all possible mutations in the gene and distinguish which are drivers and which are passengers. Now, in this case, it's drivers of clonal hematopoiesis. And this is for TET2. These are all preliminary results, but that's the way where we are going. We have not reached this, but that's kind of the plan, right? To use this also for interpretation. And the last thing I want to explain you is about the concept of another project that we are starting soon in the lab. And this, so as you've seen in our lab, we have been studying driver mutations for many years. And one conclusion that we arrived is that driver mutations are totally necessary. So we find if you search carefully enough with whole exomes, including structural variants, point mutations, and you find driver events in all tumors. Okay? So every adult tumor has one or several genomic events that drive tumorigenesis. So they are necessary. But one thing that starts being very evident is that they are not sufficient. And one of the evidence of that is this sequencing of normal tissue. This is work by Íñigo Martín Corena at the Sanger. That this is esophagus, but you also find the same thing in the skin and in other tissues, that you find emerging clones, small clones of cells with driver mutations, with TP53 mutations, NOTCH1 mutations. So these are similar concepts as clonal hematopoiesis, but in normal tissue, in solid tissue, have cells with mutations leading to small clonal expansions. And they don't develop a tumor, so they are on check. They are totally normal looking epithelium. Another piece of evidence that it was important for us to think about all that is that this is work by Alan Balmain and David Adams at the Sanger as well, and Alan Balmain in San Francisco. They tested many carcinogens that were chronically exposed in mouse. And these mouse developed tumors. And they were thinking, okay, if we sequence whole genomes of these mouse tumors chronically exposed to carcinogens, we are going to identify maybe a mutational signature or an increased number of mutations, thinking that many carcinogens are mutagens. And the surprise is that most of the carcinogens didn't show any increase of mutations or any mutational signature. So many carcinogens are not mutagens. Abel and myself wrote a news and views for that paper, and we presented like this. So in the minds of many people, including us, we were thinking that many carcinogens could cause mutations, like tobacco, for example, and many others, and these mutations are then the ones that drive tumorigenesis and clonal expansion. But now we have to think in an alternative way as well. That is that mutations that are, in any case, everywhere, because they occur from very early age every day in all our tissues. So mutations are still important, but they may be pre-existing to the clonal expansion caused by the carcinogen. And the carcinogen in some cases doesn't create mutations, but promotes the expansion of cells that already have driver mutations. Okay? This is the step that we call promotion. So instead of a mutagenic step, is a promotion step. And then there is this program that's called Cancer Grand Challenges, that they first provide some challenges, which are kind of important questions that are unsolved in cancer. And one of them is the normal phenotypes question that fitted in what we were thinking. That is how there are driver mutations in normal cells, normal tissue, and how come at some point some of them develop into cancer, but many of them do not. So the project that we wrote has this hypothesis, or that's what we try to understand, how promoters, thinking of promoters as carcinogens or risk factors that not necessarily create mutations. So they drive tumorigenesis, they change the tissue in one way or another, but without necessarily creating new mutations. We want to understand how the normal tissue is modified upon exposure to promoter that make it more prone to lead to cancer at some point. Okay? This is a very complex project. I will not explain it in detail, but for me, if I convey the message, it's basically what I aim today. We have a number of samples, normal samples from patients exposed and non-exposed to promoters. And also we have some cases where we have intervention. So we have samples before and after exposure to promoter. And so we are going to focus on the normal tissue and try to identify first these emerging clones with driver mutations, with deep sequencing, and then trying to understand how they phenotype change upon the exposure of promoter. Okay? This is the team. This is a very complex project, and we need this interdisciplinarity. Paul Brennan is an epidemiologist. Alan Balmain is expert on mouse models and carcinogenesis. And then we have other team members that are key to the success of the project. We've been working many months, 1.5 years or so online, and recently we were able to meet in Washington. And this is the team. That's the first time that we see each other face to face, and it was very exciting. So the project has now started this month, and I wanted to explain a bit the concept. So I finish here. I want to thank everyone in the lab. I mentioned, I think, everyone involved in different parts of the project, but all the team is important for all these projects to succeed. I want to thank people who generate data and they share it, because that's key for us and many other people to do other research. And I want to thank also the funding agencies. Thank you.
Thanks so much. Questions? Yeah, please.
Excellent. Okay, thank you. So this morning we were talking about how the microenvironment may affect the immune system. We were talking about other conditions. And I even mentioned that even drivers in mouse models require something extra that I named inflammation. For instance, mutated KRAS does not induce pancreatic cancer unless you induce pancreatitis, inflammation. Right. So in your machine learning stuff, I mean, you are getting closer, but I need to ask you, in your machine learning pipeline, are you implementing what is happening in the surrounding of the tumor? Are you doing something to understand whether the stroma or the immune system are allowing the tumor to grow?
Yeah, no, that's a very good point. So in the machine learning, no, so we only take into account the mutations that we have observed, and as a context is the tissue. So we can say a mutation is a driver in this tissue, but not in this other tissue. not because we think that the other things of the tissue are not important, but just because we need to simplify things so that with the data that we have, we can effectively learn. But we have in mind including other things related to the context. For example, we know that other mutations that occur in the tumor are important to define if this new mutation may be a driver or a passenger. And as you say, what happens with the context, right? How is the immunosystem around? Do we have T-cell infiltration or not? Or if the patient has been treated with chemotherapy, because there are some mutations that suddenly acquire positive selection under treatment. We have in mind with Ferran, who is here, to generate models that will take into account more context. So there are things that we can improve, but the limitation is going to be the data. So we could, so imagine in an ideal world that we have millions of somatic mutations from millions of samples, and we have also information about expression, T-cell infiltration, inflammation. This is possible, but we don't have this data. So we will advance as much as possible with the available data, but it's a totally correct point. Yeah.
Following up on this a little bit, I'm thinking of the example of liver cancer. Maybe it would be a great setting because it's very clear where you, well, if you have minimal metadata, you will have if it's a cirrhotic or not. So you will have if there is inflammation, and that would be maybe a variable. Like maybe there are some settings where typical clinical variables as etiology will have been annotated, and maybe it's a good place to start and fit the model.
Yeah, that's a very good point, because there are some of these features that will be there, so that we may not need expression in the normal tissue, but you may have cirrhotic
exactly.
Clinical event. That's right. And what we would like to be able to learn, I mean, we are a very long way. I'm telling you what is kind of my dream, but could be this, like, is when this mutation occurs in a cirrhotic patient that has been treated with chemotherapy, is this important? And maybe in this patient it is, and in this other patient that is not cirrhotic, it's not. But yeah.
And if I may, just one very specific question. When you, I thought it was very nice, the topic about the mutations depending on the transcription factor binding site. So depending on the cell type, actually, then that means, right? Have you considered crossing all these human cell atlases where there's a very specific profile of the transcription factors active in a specific cell type to maybe try to, from the cancer, understand what's the cell of origin?
Yeah, okay, that's a very good point. We are not doing that, but I know some people is, for example, from the overall pattern of mutations in the genome, you can infer the cell of origin. Because all this depends on chromatin conformation, and chromatin conformation is telling you cell of origin. So that is possible, yeah.
Okay, thanks for your talk. It's always nice to hear you. You always have a lot of interesting ideas, and it makes you go to different places. Just as a question, maybe this is just like something very puntual, no? But you were talking about how you find the drivers of clonal hematopoiesis, okay? And some of them are related with the drivers of tumorigenesis. And we're using always blood as a well, no, not always, but in most of the cases. Do you think that we are losing some of important mutations drivers because we're using blood sample as a control? Do you think that can play a role? Or?
Yeah, I mean, we know that in some cases this has been an issue, so that, for example, some of these genes, it's true, they are important in leukemia, for example, and some of them are important in some solid tissues. So in some cases it happens, and it happens also the other way around. So we have to be careful, especially with these genes, when you are calling somatic mutations in the tumor, because in some cases you may mislead that this is a germline variant. It's not that it's kind of a super big problem, but there are some issues that require extra attention because it's true you can mislead them as not like germline variants, and these are clonal hematopoiesis mutations. That's true.
Okay.
other like chromosomal amplification, and we know that that doesn't mean that you have a regulated gene, that gene can go up and down.
So yeah, no, you didn't miss it. I didn't talk about structural variants. Again, not because I don't think are important, because they are super important, but the way we model for positive selection and for in BoostDM, we can only do it in a way that we trust for point mutations and indels, especially point mutations. Because we can simulate this neutral mutagenesis, we think that quite accurately. We don't know how to do that in structural variants, copy number alterations. Cancer Genome Interpreter deals with them, because if you put MYC amplification or a fusion event, we will tell you that this is a driver event. But we are not so confident in how to learn it directly from the data. Thank you.
Okay, so we arrived to the final presentation of the day, which is Jordi Esteve from IDIBAPS. So do you know which presentation is your one? Do you know where
I will try to find. Thank you. This one. So thank you very much, Ivo, for inviting, and all the team for inviting me to participate in this meeting, especially to have the opportunity to attend to these outstanding talks. I was really astonished by the talk by Núria. I feel a bit like an alien. I am a clinician, a poor clinician, no? Among very wise people and with a high level of knowledge on genomics. And I think my commitment here is to explain you how we are implementing this information in clinical routine. I think it's interesting, and with this interplay, I think we can at least plan or to make to bring you some important issues and med needs that are important to develop in the next future. So how we implement this genomic knowledge, for example, in the management of acute myeloid leukemia. This is something that physicians do that to show our disclosures, because we have disclosures. So my learning objectives would be just to show a bit the diversity of this disease, of acute myeloid leukemia, and how we try to organize and to identify entities that has meaningful clinical and that has a particular treatment. No? And we are learning about the biography of the disease, which is something interesting. No? Núria mentioned clonal hematopoiesis, which is an important event in this disease. No? How we are moving from intensive toxic chemotherapy to targeted therapy, I think that's something very important. And other concepts such as what we call measurable residual disease, the way we try to assess in a deeper way the response to therapy, and what is about immunotherapy in this particular disease. Well, Núria mentioned, and many of you, AML is a disease with a relatively low number of mutations compared to other tumors, such as melanoma, gastrointestinal, or, for example, lymphoid neoplasm. That's something that hematologists treat. We know the more important mutated genes in this disease. It's a relative limited number of genes. It's important that we know the functional implication of these mutations. We can classify mutations in 9 categories. It's also important to see gene-gene interactions, how some mutations cooperate. No? For example, NPM1 with FLT3, probably mutations in signaling pathways are necessary to develop a mutation in a patient with an NPM1 mutation, and also mutually exclusive mutations. No? And with all that knowledge at this moment, we are capable to at least recognize 13 individual entities defined by a molecular event. No? These are listed here. Very recently, two different AML classifications have been published, have been presented. No? They are coincidental in most issues, but to have the two different classifications is not the best scenario. No? And this classification proposed something that I think is interesting and smart. It's a hierarchical way to classify leukemia. First, we interrogate if this particular AML has any of these 13 entities. If not, it's very important, it's relevant from the clinical standpoint, if the AML has a mutated TP53, because this will determine prognosis therapy. And at this moment, it's very important to identify genetic or gene mutations or chromosomal events that are related with myelodysplastic syndrome. And these gene mutations or these chromosomal events will assign that particular case to AML with myelodysplasia or related with myelodysplasia. I should mention at this point that we have an ongoing project to classify, to understand different entities in those patients that have not a genetically defined AML. This is something that is ongoing. Chisca Guijarro in my group with some of you develop, and we have RNA sequence of 100 cases, because this is not probably a homogeneous entity, but an entity that comprises different entities. And if none of these genetic events is found in that particular case, well, we should admit that we are not able at this moment to classify that particular AML with genetic tools. Going to the cell of origin, well, the cell of origin of AML is in most cases a hematopoietic stem cell that is transformed by the acquisition of some mutations. But we know that a minority of cases probably are originated in a T cell progenitor. And this sometimes it's confounding, no? Because we find a mixed phenotype, and in that particular case, the genomic program is more related to AML. So in some cases, the cell of origin is not the typical one. It's a cell of origin reprogrammed with typical myeloid mutations. Well, when we perform NGS at diagnosis, this is a static picture of the case, no? Because in most cases, that particular disease comes from clonal hematopoiesis of many years ago, no? And with that expansion of a normal, healthy clone, with the acquisition of additional mutations, develop finally an AML, no? And that AML will be treated usually with chemotherapy. In many cases, we can induce a response, but we know at this moment that most of these responses, or at least an important part, a large part, are clonal remissions, are not, the patient does not recover a polyclonal, completely healthy hematopoiesis. And after some years, this clonal remission can induce a relapse, but sometimes a novel clonal evolution, and something that I will show you in a few minutes. This is a real case of a patient treated at our center. The patient was diagnosed with an AML with NPM1 FLT3 mutations. These are typical hallmarks of a frequent AML subtype. The patient was treated with chemotherapy. She achieved clonal deep response, and after some years, they developed thrombocytosis. And at that moment, when we performed another NGS, we saw the emergence of a JAK2 mutation and NF1. And she was finally diagnosed with a polycythemia vera with the acquisition of JAK2. It's interesting because there is a change in phenotype, there's a change in disease. And this change is related to the persistence of a clonal hematopoiesis in DMT3A in that particular case. And over that clonal hematopoiesis, there is a higher risk to acquire a second myeloid neoplasm. It's important because depending on the phenotype, of course, the treatment is completely different. No? With AML, we should intervene immediately with chemotherapy, in many cases with hematopoietic stem cell transplantation. Polycythemia vera is another disease with a higher risk of thrombosis, with somehow potential of transformation, but with very different. No? This pattern of clonal evolution is more and more frequent. We are detecting a lot of those patients, especially patients that do not undergo allogeneic stem cell transplantation. And that's something we are interested in, and we are, these days, we are trying to develop a project of single-cell sequencing just to track this clonal evolution a long time. No? Núria mentioned the concept of clonal hematopoiesis. That's something fascinating. No? We can find in normal individuals, or at least without a hematological disease, this kind of mutations, typical of myeloid neoplasms. The most typical and frequent are DMT3A, ASXL1, and TET2. And we know that these individuals have a higher risk of developing a myeloid neoplasm, which is, in a sense, is quite logical. But there is also a known risk of a higher cardiovascular events, and it's something important, probably because these mutations induce a higher atherogenic potential to the vascular atherogenic plaques. No? And if this is finally confirmed, that's something that in the future can, for example, promote the investigation of CHIP, of this clonal hematopoiesis, just to prevent, for example, the risk of cardiovascular events. Let's move to treatment. The current paradigm of AML treatment is simplistically divided in the possibility to cure a fraction of patients, younger patients, using intensive and toxic chemotherapy, and in many instances, allogeneic stem cell transplantation, especially in those patients with a high risk of relapse. Well, this is a treatment that it can be used in younger individuals, but it has a higher toxicity. It's not possible in older patients. No? And for those patients, we did not have any effective tool, any curative tool, until very recently. And we developed a lot of combinations that we'd call as low intensity. Low intensity as meaning that they have myelotoxic potential, but a lower toxicity, extra hematological toxicity profile. But at this moment, we have very useful combinations using targeted agents such as FLT3 inhibitors, IDH inhibitors, or venetoclax, which is a BCL2 inhibitors, that produce or provides a high response rate. And these low-intensity regimens are now being transplanted to younger patients. At this moment, these are real results in patients treated in our cooperative group with a treatment based on intensive chemotherapy and allogeneic stem cell transplantation. We can distinguish perfectly according to their genomic profile in let's say, good prognosis, intermediate prognosis, and high or poor risk prognosis. All is relative because, well, in good prognosis patients, you lose 20 or 30% of patients in 2 years. No? And even more, when we analyze the poor risk category, we are capable to identify a group of patients that are at this moment at clinical unmet needs with a less than 20% chance of survival at 2 years, despite performing high-intensity chemotherapy regimens and even providing them allogeneic stem cell transplantation. These are patients or AML characterized by TP53 mutations, usually associated with complex karyotypes, but also this particular rearrangement that leads to a rearrangement of the MECOM gene with an enhancer of the GATA2. So this insufficient or this frustrating reality is what warrants the development of targeted therapy. No? Considering targeted therapy, we have to analyze the adequacy of the targeted therapy we are using. No? The frequency in AML, no? If we are targeting a mutation, how recurrent is this mutation? The specificity in AML cells, this is something essential, no? Because the toxicity is mostly derived by this on-target toxicity. And also it's very important the order in the leukemogenic process. No? If it is found there an initial event, probably the target therapy will be more useful as compared to an agent that is targeting a subclonal, more evolutive event. No? It's also interesting to know if that targeted therapy has a synergistic effect with known effective current therapies, with intensive chemotherapy or, for example, with hypomethylating agents. Or, for example, if there is some kind of interaction of synergy with the allogenic effect obtained after allogenic stem cell transplantation. We know that, for example, using some agents after allogenic stem cell transplantation, we can induce more durable responses, just because there is a synergy, there is a collaboration between the immunogenic effect of the donor lymphoid cells and the targeted agent. Of course, it's very important when we use targeted agents, and we see that the loss of control, to analyze the mechanism of resistance, the escape, how does cells escape to the control, and part of the relevance of a targeted therapy is to analyze the off-target effects, which are mostly rely on the specificity of the agent we are using, and those, let's say, on target of tumor. That depends on the specificity of how distinctive that target is expressed in leukemic cells and not in normal hematopoietic cells. So at this moment, we have several targeted agents that are useful, are being approved, and are used in AML, such as FLT3 inhibitors, which is a tyrosine kinase inhibitors. We have a specific formulation of chemotherapy, liposomal. We have BCL2 inhibitors that promote apoptosis. Also hedgehog inhibitor, or something like the trioxide, arsenic trioxide, that allows the degradation of a leukemogenic initial event in polymonocytic leukemia. And of course, we need to develop new targets because we have a lot of unmet needs. So at this moment, when we diagnose a patient with AML, we perform this kind of routine analysis. If the patient has a FLT3 mutations, because he will benefit from a FLT3 inhibitor. If the patients have a core binding factor fusion gene, because we know that combination of chemotherapy with a particular conjugated monoclonal antibody that's called Mylotarg gemcitabine, it's useful. Or, for example, in patients with AML related with myelodysplasia, can benefit from the use of a specific and new chemotherapy formulation that is based on a liposome. Apart from that, we know that patients harboring IDH1 and 2 mutations are sensitive to specific inhibitors, but also are very sensitive to the combination of venetoclax with hypomethylating agents. And what happens when we find and we diagnose a patient with TP53? This is really a very challenging situation. There is no one effective therapy, chemotherapy, nor allogenic stem cell transplantation. Those patients tend to relapse after allogenic stem cell transplantation, nor low-intensity regimens such as venetoclax. So for those patients, I would say that at this moment, we should prioritize the inclusion in clinical trials with novel drugs, because it's the best chance to get a meaningful response. No? And there are other AML subtypes that can benefit from ongoing trials. Examples of that, the use of midostaurin, which is a FLT3 inhibitor, in addition to chemotherapy, provides a survival benefit. Those patients relapse less, and there is a proof of concept, and this led to the approval of the drug, that the combination of intensive chemotherapy with that particular FLT3 inhibitor results in a better outcome. Moreover, gilteritinib, which is another FLT3 inhibitor, is a second generation, more specific, in monotherapy in patients relapsing after frontline chemotherapy, is better than any chemotherapy that we can use in patients with mutated FLT3 AML. These are the good news, but the bad news is that the response is limited in time, no? In most patients treated with gilteritinib. although there are some AML subtypes that can benefit from these specific combinations, such as patients with commutations in NPM1 and DMT3A, we know that patients treated with gilteritinib after some months will relapse, will progress, and when we analyze these progressing samples, some patients will lose the target, no? Some patients relapse without the FLT3 mutation, and the most recurrent cause of progression is due to the acquisition of mutations in the Carreras, PTPNO, BRAF pathway. So this is important because with that we can design, we can implement, we can think of a combination that tries to avoid this mechanism of escape. Well, now we are performing routine NGS in patients diagnosed with AML, and with routine NGS we are observing, finding every day novel variants. That's something that Núria addressed. And for some of these variants, which are unique, we don't know the particular sensitivity of FLT3 inhibitors. No? We can infer, no? Depending on the region they are located, but something is a knowledge that we need to create just to know if that particular inhibitor is useful, or we should apply another inhibitor. No? Another important mutation that also has led to the development of targeted therapies is IDH mutations. IDH is a protein of the Krebs cycle. Probably you know much more than me about the Krebs cycle, something that I studied many years ago, and I thought that never in my life I would meet again with the Krebs cycle. I was wrong, no? Because in AML, treating patients with AML, I should learn. Well, that particular mutation generates the production of a metabolite that is called 2-hydroxyglutarate. This metabolite can be even found in blood samples. It's something that has been performed in the biochemistry department of our hospital by a sensitive technique, gas chromatography with mass spectrometry. And that particular metabolite generates epigenetic dysfunction with a hypermethylated status in DNA, but also in proteins of the nucleosome. No? It's important because it's a frequent mutation in AML. About 20% of patients harbor one of these mutations and can be targeted, and relatively safe because it's a specific AML cell mutation. No? Very recently, we have read this interesting paper that in older patients which are not candidates to intensive chemotherapy can benefit from the combination of ivosidenib, which is an IDH inhibitor, specific inhibitor in association with azacitidine. And the results, those patients have a higher rate of responses compared to the standard of care until this moment that was azacitidine. And those patients are alive, 50% of these patients are alive after 2 years with a low-intensity regimen that is very well tolerated. Well, you might say that it's not so good news, but just to remind you that before that, the median survival of those patients was less than 1 year, was about 9 months. So this is an important and a strong signal of the sensitivity of this AML subtype to this combination. Venetoclax is a very important, a very active agent in AML. Combined with hypomethylating agents, has a selective effect, which is interesting because it's a safe and low toxic combination, and has transformed the management of older patients with AML. The pivotal trial is the VIALE-A, and the combination of venetoclax, which just to remind that is a BCL2 inhibitor, results in a higher response rate of about 2/3 of patients treated with this combination, meaningful or clinically meaningful prolongation of survival, and interestingly, about 1/3 of these patients are alive after 2 years. Moreover, we know that some specific genetic marks, such as the presence of IDH mutations, determine a high sensitivity to this combination. And patients with, for example, IDH2 mutations treated with this combination have and median survival not reached at this moment, which is something that has revolutioned the field. Well, venetoclax is the cornerstone of the treatment of older patients, of low-intensity regimens, but unfortunately, most patients treated with venetoclax will progress after several cycles of treatment. And when we try to analyze the causes of this progression, we see that emergence of some mutations, for example, TP53 mutations, determine the loss of effect of the drug, which in part is logical, because it's a very anti-apoptotic signal. But also other mutations that promote or prime other ways of alternative apoptosis based on MCL1 or BCL-XL. And those patients, or the emergence of these mutations, make that venetoclax is no more useful and clinically active. So we need to develop novel agents, novel combinations, especially novel combinations. For example, we need to develop IDH inhibitors, which can be useful for both kinds of mutations, IDH1 and IDH2. Some of them are being experienced in clinical trials. We are also experiencing with bispecific T-cell engagers antibodies, and antibodies that attract T-cells against the particular target that is expressed in the AML cell. We have some interesting novel targets, such as menin inhibitors, that are especially expressed in AMLs with KMT2A rearranged subtypes or patients harboring NPM1 mutations, and other targets that are of interest and at this moment are the rationale for clinical trials. Beyond specific individual mutations, we could analyze the genome or the gene expression profile of patients and to just try to correlate which are the genome profiles that are associated to a specific response. And something interesting, this was performed in an interesting trial in the United States that was BEAT AML. And with the information generated by this trial and many others that can generate in the future, I think probably this will be something useful in the future to just to make the best choice for a particular patient. Well, the concept of measurable residual disease refers to the persistence of a, let's say, low load population of AML that is responsible, it's the cause of the relapse of the patient and a proportion of or a population of AML cells that is not detected with conventional tools. This is something important because if we detect that MRD persistence after an intensive treatment, maybe we can implement some kind of additional treatment maintenance or MRD driven. We know that the persistence of MRD has a clear clinical prognostic impact. And a nice example of that is an example of a patient treated in our center harboring a RONX1 mutation. After intensive chemotherapy, although we thought that he was in remission because we do not see morphologically leukemic cells in the bone marrow, by performing this technique of a droplet digital PCR, the mutation was persist, and the reduction of the leukemic burden was really very low, of 2 logs. But after performing allogeneic stem cell transplantation, we could eradicate, and that's something interesting, and we can follow that patient. And if there is a reappearance of the mutation, which is characteristic, it's associated with the leukemia, maybe we can implement an immunological strategy just to revert the situation. So MRD tracking is something very interesting. It's based on genomics, on the profile of the patient, and it's something that perhaps can allow us the development of preemptive strategies. An example of that is another real case of a patient that was characterized by something very particular, that was the coincidence of two gene fusions, core binding factor with this is the gene fusion characteristic of inversion 16, with BCR-ABL, that's the characteristic gene fusion of Philadelphia chromosome. Well, that patient was first treated with chemotherapy and a targeted therapy, dasatinib. We could eradicate the BCR-ABL, but the inversion 16 or the core binding factor beta with the myosin heavy chain 11 persist. So meaning that this patient was at risk of relapse. We tried to, the patient received an allogeneic stem cell transplantation. We tried to promote, to enhance the immunological effect by giving lymphocyte infusions of the donor. With that, we achieved some kind of reduction. But after some time, unfortunately, the patient present a clinical relapse at CNS. She was treated. After several months, there was another relapse. And at that particular point, we started with a low-intensity regimen with venetoclax and decitabine. And for the first time, we have been able to clear completely this molecular marker. This, I am not completely convinced that it's definitive, so we can follow or we should follow the patient. But it's an interesting story of combination of targeted therapy, intensive chemotherapy, allogeneic stem cell transplantation, donor lymphocyte infusions, and at this moment, venetoclax with azacitidine. Let's say just a few words of immunotherapy in that field, in the field of AML. Well, the first proof of immunotherapy is the use of allogeneic stem cell transplantation. In fact, allogeneic stem cell transplantation, or the first indication of allo transplant at that moment, is AML, which is continuously growing because it's useful. It's interesting because with that, we prevent relapses. We apply and we use allo transplant, which is a complex and toxic procedure for those patients with a higher risk of relapse after chemotherapy. And we know that there is an imbalance, and there is an interaction between donor lymphocytes and AML. And that's demonstrated because when we analyze samples of patients relapsing after allogeneic stem cell transplantation, in some instances, we show that blood cells, the leukemic cells, lost the sites of recognition of the allogeneic effect. That is the HLA class 2 antigens in the surface. So the leukemic cells downregulate the expression of these markers and avoids the immune recognition by the donor lymphocytes. So that's something interesting, no? And something different from clonal evolution after chemotherapy. Well, you know that there is now a big interest in the development of CAR, of chimeric antigen receptors in autologous lymphocytes. This is a very useful strategy in some lymphoid neoplasms, in acute lymphoblastic leukemia, in lymphomas, in CLL. No? These are very sensitive tumors, but there is no such a success in the field of AML. So for that reason, we are trying to implement with the help of Pablo Menendez's team an academic CAR against AML. And for that, the first choice is to decide which is the best target. No? And we choose CD123. This is part of the receptor of the interleukin-3 receptor. This is a specific, it's highly expressed in AML samples. It is almost exclusively expressed in hematopoietic stem cells, which is something interesting. And we have developed our, with several hybridomas, we have developed our specific academic construct, and this construct has been has been tested preclinically and shows an interesting activity in cell lines, also in primary AML samples, and in murine models such as this one. The CAR-123 has the capability to avoid, to abrogate the expansion, the growth of an AML infused in those animals. And with that background, we are trying now to escalate to the clinical use with the hope to obtain at least results similar than those obtained in lymphoid neoplasm. And with that, I would like to finish some conclusions about the implementation or the translation of genomic knowledge to clinical. Just this genomic characterization allows us a more accurate diagnosis, and this allows us a refined prognostic assignment, a better prediction. We can predict the response to treatments we are using, identification of targets, of druggable targets. This is something that we do every day after a relapse. They provide us markers for measurable residual disease monitoring. They also provide us hints to track clonal evolution in some patients that make this kind of change of phenotypes. At relapse, allow us to investigate if there is a treatment that we can use with some hope of obtaining a response. And also, it's something that we have not treated in the particular talk, but we can identify the presence of germline predisposition mutations with something relevant. And also, genomics impacts therapy by development of targeted agents, such as those examples I have explained. We also, with this kind of research, will allow us to investigate the mechanism of progression on how to use combinations in a more useful and more effective way. We have an urgent need to develop effective therapies for our high-risk AML subtypes. And also, the tracking of MRD can warrant the use of preemptive strategies before an over-relapse. And with that, I would like to thank all of you for your attention. I hope you are not bored for this clinical talk. And of course, to thank all the people that is working in the clinic, also in the lab, in the cooperative groups, the LAM, and all the research teams that are helping us a lot, such as the Pablo Menendez one that I have mentioned before. So thank you very much.
Very good. Thank you very much. Do we have questions?
Thank you very much for the talk. Really enjoy it.
At least someone did.
So I will go first maybe for the IDH2. I'm not sure if there are any mutations status in MYC, but what we have observed, I mean, actually what I would like to know if MYC could be overexpressed or somehow altered in AML, because what we have observed is that MYC, and in our case MYC-N, regulates IDH2, specifically IDH2. And although it is not mutated, overexpression may somehow push up the levels of 2-hydroxyglutarate, and that's...
So I must confess I am not an expert on that. I would say that mutations in MYC are infrequent, but are found. In fact, the group of Eli Papamanuil, which is someone who has worked a lot, found a group of AMLs with characteristically MYC mutations. I am not aware of some correlation of the MYC expression with patients with IDH2, but I will investigate on that. It's an interesting question.
And another thing is about the clonal hematopoiesis being associated with cardiac. Is it the chemotherapy or no? It's the clonal expansion.
The first hint was just an epidemiological one. No? When those papers just identify and reporting the phenomenon of the clonal hematopoiesis, they see that patients with a chip had a higher risk of mortality. And the first impression is, well, this is due to the development of myeloid neoplasm or hematological malignancies. But it was not the case or the only case, because those patients had a higher mortality by cardiovascular diseases. Well, the first impression is that this is just some serendipity finding, but it does not seem the case because focus studies, retrospective and prospective, have seen an association with a higher risk. Patients that develop a myocardial infarction has a higher risk to harbor a CHIP, a clonal hematopoiesis, in JAK2 especially, but also in TET2, in the MT3A. And the hypothesis, there are some murine models just trying to see what happens with clonal hematopoiesis. And the hypothesis is that perhaps those cells deriving from a clonal hematopoiesis, monocytes are part of the serum plaques, no? So there is a higher expression of cytokines, but also monocytes that are derived from a chip can have a higher heterogenic potential. This is the hypothesis. The epidemiological relationship is now clear, I would say, and the hypothesis, the biological hypothesis is ongoing. It's something interesting.
Thank you.
Okay, any other questions?
Just one detail. You mentioned at the beginning that there were different cells of origin that were identified. Are those based on histopathology, so the morphology of the cells, or is it based on the expression of the cells that, because you said it as if it's something that it's known and described?
Yes, this was something, when we analyze the phenotype of the cellular disease, most of leukemias have a myeloid phenotype with no expression of other lineage markers. No? But we know that some cases that were considered biphenotypic or mixed lineage, harbor T-cell markers in association with myeloid markers. And those cases, when you analyze the mutational landscape, is a typical myeloid one. No? So this prompted to consider that perhaps some AMLs come from a common progenitor, something that is physiological, that acquires myeloid mutations. No? So this kind of evidence comes from the mutational landscape, also from gene expression. No? But it's not so static model, but the cell can reprogram and can have, depending on the acquisition of the mutations, can have some kind of transforming or expressing another lineage.
And then do these have different clinical behaviors, or is it like, are they include, like these clinical trials do include all of them, or, you know?
This is a challenge because we tend to classify cases according to the phenotype in a very systematic, if you have T-cell, and this has led sometimes, I think, to make some mistakes. No? And we know that those very early T-cell leukemias do not respond well to typical acute lymphoblastic leukemia chemotherapy. No? And with that knowledge now, we are more and more giving importance to the mutational landscape. No? And perhaps the cause that was that the program of that cells is not sensitive to typical ALL chemotherapeutic agents. And now we treat those patients with more myeloid schemas. But something important. No? So we have to integrate all this information, because if not, you can rely too much on the phenotype, for example.
Thanks so much, Joe.
Thank you.
Okay, good. Yeah, we've reached 4 o'clock, and I want to thank again all of the speakers for their willingness to come here in person and give a presentation. That's much appreciated. I want to thank you all for participating. I want to thank Illumina for the funding that they gave us. The nice lunch came from Illumina, more or less, and yeah, so it's been an absolute pleasure to have you all here, and to be here in person again is great. There is a tour of the facilities that people could sign up for. Those of you who signed up for the tour of the facilities should just wait in front of the auditorium. There will be people who pick you up and take you to 2 different places, I think. Show you a computer and show you loads of sequences, I believe. Okay, thanks ever so much, everyone, and yeah, I'll see you maybe at the 12th one of these symposia. Okay, thanks a lot.